ओके, एक्चुअली हमको इसको यहाँ से नहीं, ब्राउजर से, वहाँ अकाउंट से वो लाइव जाना था। तो नहीं भाई, स्पीकर ऑन है?
स्पीकर ऑन है सर, म्यूट कराना है।
हाँ,
एक बार चेक करो, फिर लॉगिन नहीं, आप एक बार चेक कीजिए।
उसको यहाँ से नहीं, ब्राउजर से, वहाँ अकाउंट से वो लाइव जाना। हाँ तो
जूम में
स्पीकर ऑन है?
नंदी सर नमस्कार, सैकत जी नमस्कार। नंदी सर नमस्कार।
हाय, गुड मॉर्निंग सुरेंद्र, कैसे हो?
नमस्कार नंदी सर नमस्कार। Some
There is too much of echo here. I don't know why.
echo is maybe I will need the क्या हो गया? Sir, pranam. Sir, pranam.
Hi, Shuvendra.
Sir, pranam, sir, pranam.
Good morning. Can you see my video?
We are not able to.
Video is not clear, is it? Video you can't see?
I can, sir. I am able to see your face.
Okay, okay.
Yeah.
Is it clear or not clear?
It is clear, sir.
Okay.
It is clear. It is
Okay.
clear.
Great, great.
Good morning.
Madam, namaskar.
Good
Hi.
morning.
Hi, Subra. How are you?
Hi, I'm fine. How are you?
Doing well. Doing well.
Yes, so your slides are very, so something is there which I can see very well.
Yeah, I'm going to put my slides now.
Okay.
My slides.
So I thought I'm late actually. So,
Yeah. No, I think
good.
9:30 is the meeting.
Yeah, okay.
So, yeah.
But in the program, it was 9 o'clock, no?
Oh,
9:30.
today
Subra.
is 8:30.
Okay, okay, good.
9 is on tomorrow's meeting will because of the US time.
Huh,
We are starting at 9 tomorrow.
that's fine. That's fine.
Namaskar, namaskar, Subhadeep.
Namaste, namaste, Subhendra. So maybe I can just for 5 minutes, then Subhendra can take over.
Sure.
So I welcome all for this MIPM 2022. On the occasion, this is also our occasion of the CDFD Silver Jubilee celebration, which our director, Dr. Thangraj, also was very encouraging and also induced us to have this meeting. So initially, some time back, we thought of having a physical meeting, but unfortunately, because of the changing situation, so we shifted it to an online mode. And we were very interested in hosting all of you in Hyderabad. Even we have started looking for guest house and other arrangements also, and we got some sponsorship also. So anyway, like it's good, like we will have very good session, I believe, online sessions. And then in the month of May or June, we will plan it again. And if based on that situation, we will again plan to have a physical meeting in the winters in 2022 or early 2023. So with that all, I welcome you all and thank you, Ramesh, for agreeing to give our plenary talk. We will have plenary sessions all the 3 days. So for tomorrow, Steve is giving, and day after tomorrow, Shubhra will be giving her talk. So I will not waste time and hand it over to Suvendra. So he will introduce and will start the session.
Namaskar.
Namaste.
So actually, we are all grateful to Subhadeep for putting so much of effort, and sir will be also feel very pleased to see that how the MIPAM Subhadeep has taken to one more level, and so we are grateful to you. Then in the last part, sir will be talking about. So in this MIPAM, we take sir's philosophy of patriotism and also sir's philosophy of doing science, which you observe and speak. The quality science, what we have learned from him, we are trying our humble effort, and it's because of his kindness and blessings we are able to organize the MIPAM, and his unconditional affection, support to everyone who is in science and who is for the country. It is always we try to follow. So actually we are only trying our best to follow the philosophy. His philosophy of respecting everyone and learning from all. This is the way we need his blessings. So everyone who are part of this or listening, they will realize that or they know. So I have will just listen to Sir. So more we listen to him, more we learn. So with this, I think we will start the talk by Sir. So Pranam,
Yeah,
Sir.
Arun,
You should start your talk and we will be listening to you, sir.
Sure.
I think we are a little early because you're supposed to start by 5 minutes later. And
Okay.
you can see more people would maybe join in a few minutes, I believe.
Maybe I'll share my presentation.
Yeah, it's good. We can start at 9:35, another 2-3 minutes.
Yeah, I'll just
Right.
share-
Actually, I'm sure many
Ramesh
are
can
waiting
load
to
his
hear
slide
Ramesh.
yeah.
I'll just
I
share
think
my
that's
presentation.
Yeah, I also think so, because I don't see many of the students in the crowd actually still.
Yeah.
So we
No,
can
no, they
wait.
will come. I think
Yeah.
nobody anticipated to have little early start. No?
So it's okay, but
Maybe
can you
5
see my
minutes.
presentation? Can you
Yeah.
see my presentation?
Yeah.
Okay, great. Yeah, we'll wait for a few minutes.
Yeah. Around 5 minutes we can wait.
Yeah, no problem. I think it's really nice to have this meeting. I mean, the original idea was that we all get together to hear
Get
about,
together, yeah.
to hear about plant microbe interactions, exchange ideas in person. Anyway, COVID has intervened and we can't do it. We will have
Actually,
another-
we were very keen on having this thing in Hyderabad, and Thangraj also agreed, and we all arranged.
Yeah.
And even Hitendra started booking the guest house also, and we have got some good sponsorship also. So, but unfortunately, because of that, of the situation, this thing.
You see, now it has become human beings propose, COVID disposes.
Okay.
So we don't know what will happen and how,
Yeah.
what the future of the pandemic will be. We don't know. So
Actually, many people are getting now this infection. Even 2 weeks back, I think we all had that infection and went away in our home.
I see. Yeah. No, my mother had COVID just last week actually, but it was very mild. I think people who are vaccinated, it's pretty mild.
Yeah, it's getting milder.
No, Subra also had. Subra had it, I think, right? Subra?
Yeah.
Many people have had it. Luckily, so far I have escaped it.
No,
I think myself, my wife, and children's, they had a Omicron-like symptom 2 weeks back, but doctor told if you do the test or don't do the test, it doesn't matter. So just stay in your home for 1.5 weeks and then come. So now we have recovered, and Dr. Thangraj also last week had COVID.
I see. Yeah, very tough.
No, actually, both me and Niranjan got it on 1st January, but that is because of all these 2 years I protected myself like anything, because my mother is with us. She's almost 84. But then that was our mistake only. We invited somebody, it was a Saturday, and Thursday evening I invited someone who came from Calcutta thought that it will be all okay, which I have never done. I actually opened my mask and had, dinner and served dinner with them. And
Yeah,
immediately I got it because they were traveling out from Amritsar and then coming to Delhi. And one of them had it. So basically it was my mistake. Otherwise, I think if we have mask on, everything is okay.
True.
But Subhra, this variant particularly, even in the labs, we are wearing masks, etc. And in the like outside also, I am wearing mask. I don't know how I got.
Somewhere, Shubhodip, it's my thinking, what I'm observing. Somewhere, if you are opening the mask and having some food, that is the main problem.
Yeah, that's maybe the.
That is
I'm
the main
having
problem.
food in the canteen.
That might be the source actually.
That might have caused.
No, you can be careful for so long. After some time, sometime you let your guard down.
No, it was more of a, your own relative, and you think that they're all okay. Something like psychology comes, no? So that happened. But otherwise, I am taking my lunch for last 2 years in this room only. I don't go home even. So in that case, I open my mask when I am taking food, or now that nobody is here and I'm in this. So otherwise, I'm on mask completely. So therefore, when I took that lunch every day in this room, or my tea here in this room, nothing happened in last 2 years.
Yeah.
I think it's 9:35 now.
Yeah, we can start.
Okay. Yeah. So I'll put it on full screen.
Yeah.
Yeah. Can you see it?
Yeah.
Yes.
And moving?
Yes, sir, we can see.
Yeah, we can see.
Yeah. Now, thanks everyone for joining, and thanks to Subhadeep for organizing the meeting, and Suendra for that kind introduction. But he embarrasses me actually. But I think all of you, the whole community of plant microbe interactions community in India is doing extremely well. And I'm just really happy to be a part of it. So today I'll tell you a little bit about some work that we've been doing on cytokinin as a bacterial signaling molecule. Now we all know about cytokinin as an important plant hormone. We also know about bacteria that associate with plants and produce cytokinin to influence plant physiology. But we never really think of cytokinin as something that can influence bacterial physiology itself. Okay, and today we will talk about one particular bacterium that makes cytokinin, and the cytokinin that it makes actually influences bacterial physiology. And in the end, I will just mention very briefly that we think that cytokinin as a signaling molecule may be quite widespread in the bacterial kingdom. So the bacterium that we work on is called Xanthomonas oryzae, Pathovar oryzae, or XOO for short. And it causes bacterial blight, which is a serious disease of rice. Now, today's talk is about a particular protein that I'll refer to as ZOQ, which is essentially a Xanthomonas protein that is secreted out of the cell, and it is secreted through the bacterial type 3 secretion system into the rice cells. So this is work that other people have done. They've shown that ZOQ is made by XOO, and through the type 3 secretion system, it is secreted into rice cells. Now, this ZOQ protein, it's around 400 amino acids long. It has two sequence features. One sequence feature is related to certain motifs that it has that allow it to bind to eukaryotic, in this case, rice 1433 proteins. And it has a second activity, which is biochemical. It has the sequence is actually very homologous to what are called phosphoribose hydrolases. And a phosphoribose hydrolase is actually the last step in the cytokinin biosynthetic pathway. So both of these features actually have functions are involved in ZOQ function. And what I will tell you today, very briefly, about the function of ZOQ inside rice cells. So ZOQ has a function inside rice cells. It also has a function inside XOO cells. And inside rice cells, ZOPQ functions by binding to rice 1433 proteins, which appear to be key players in elaboration of rice immune responses. And once it binds to these proteins, it suppresses host rice immune responses. So that's the function inside rice cells. And very briefly, I'll touch on that. And most of the talk will be about the function of ZOPQ that is inside XOO cells. And as I was just mentioning, ZOPQ catalyzes the final step in cytokinin production. And what I'll show you and try to convince you is that cytokinin is a xanthomonas signaling molecule that controls bacterial transition from a biofilm to a planktonic state. We think that this transition has a very important role in virulence of XOO. And this work has not yet been published. It's under revision, but you can see it in BioRxiv. Now, we'll start off with the function of ZOPQ inside rice cells. And all of you know that the plant cell is a formidable barrier for microbial pathogens. The cell wall actually is a formidable barrier for microbial pathogens. The nutrients are all locked up inside the cell, and the microbe has, and in this case XOO, has to break down the cell wall to be able to cause disease. Now, many years ago, Suvendra actually started this work. He showed that XOO secretes a number of cell wall degrading enzymes, and that these cell wall degrading enzymes, their role in virulence is to degrade the rice cell wall. And then Gopalji and Rajeshwari Ramanan in my lab showed that the damage to the cell wall caused by these cell wall degrading enzymes induces rice immune responses. And Gopal also showed that these rice immune responses are suppressed by XOO using effectors that XOO secretes into rice cells using the type 3 secretion system. So cell wall damage induces immune responses, and those immune responses are suppressed by XOO using the type 3 secretion system. So this is what Gopal had shown. And then again, this is the work of Gopal actually. When we say that cell wall damage induces rice immune responses, what do we mean by it? What are the assays that we use for looking at whether an immune response is induced? There are a variety of assays we use. Just a couple of them I will show you. Now, this is the rice leaf into which we inject a buffer into the midvein. Into another rice leaf here, we inject a little bit of the cellobioside enzyme, which is a cell wall degrading enzyme that is secreted by XOO. So buffer treated leaves and cell wall degrading enzyme treated leaves, and both sets of leaves are inoculated with XOO. We inoculate the midvein because that is where the bacterium grows in the xylem vessels. So we are directly introducing XOO into the infection port. Now, as you can see, the rice leaves that are treated with buffer, you can see this brownish lesion that extends down the leaf. The rice leaves that are treated with the cell wall degrading enzyme, either there is absolutely no symptom as we are showing here, otherwise you have symptoms that appear, but they are shorter than the ones that are seen in the buffer treated leaves. So there is functional resistance that you get by prior treatment of rice with a cellobioside enzyme. So this is a functionality that probably comes about because of a lot of different biochemical activities that occur within the rice plant. Now, one of those activities is the deposition of a compound called callose. Now, callose is a polysaccharide produced by plants in response to microbial infection. And at the point of infection by the microbe, the cell wall is damaged. And what plants do is that they deposit callose at the point of attack. Callose is deposited to sort of seal the gap that is being produced. So it is strengthening the wall. And callose deposits also have some antimicrobial compounds, and it is considered as a hallmark of a plant innate immune response. And if you infiltrate cellobioside into rice leaves, you get all of these bright fluorescent spots, which are callose deposits. If you want, we can discuss what the assay is, but these are the callose deposits. And buffer treated rice leaves also have some callose deposits, but are much fewer. And these callose deposits, we think, are produced because of the wound response. So treatment with cellobioside induces callose deposits. It also induces a number of pathogenesis related proteins, ROS production, etc. that I will not be talking about today. So after Gopalji finished his thesis and moved on, another PhD student, Deepanvita Sinha, joined the lab. And in a nutshell, I am telling you what Deepanvita actually showed. She tried a number of different XOO type 3 secretion system secreted proteins. She cloned them into agrobacterial tDNA vectors and introduced them into rice cells in a transient assay. So the experiment was, you treat with the cell wall degrading enzyme alone, you get callose deposits. You treat with cell wall degrading enzyme along with an agrobacterium, which is actually transferring each one of these type 3 secretion system secreted proteins. It was about 16 different proteins at that time that we had as targets. And why we had these 16, if you're interested, we can discuss later. But just think that right now, out of the 30 different proteins that XOO secretes into rice using T3SS, we had about 16 that we thought were likely candidates for suppression. Of these 16, what Deepanvita showed is that 4 of these T3S secreted proteins called xanthomonas outer protein N, xanthomonas outer protein Q, or ZOPQ, ZOPX, and ZOPZ. So 4 proteins, each one of them when individually introduced into rice cells can suppress cell wall damage induced innate immunity. Again, how that happens, we will not discuss for the other proteins, we will only talk about it for ZOPQ. So this was Deepanvita's work, and this is just an example of how she looks at these assays. So the cell wall degrading enzyme treated leaves have lots of callose deposits. If you treat with this cell wall degrading enzyme along with an agrobacterial tDNA construct that has ZOPQ, and we can show that ZOPQ is getting expressed inside the rice cells, you can actually see that the number of callose deposits are significantly reduced, and quantitation of that I'll show you in a little bit. So this is the kind of assay that Deepanvita used. She tried these 16 different proteins and found that 4 of them in this assay, when introduced into rice cells, suppress callose deposition. So, and there were some other assays for suppression of innate immunity. In all of them, these 4 proteins, but not the other proteins, would suppress rice cell wall degrading enzyme induced rice immune responses. So what I will talk to you right now is about the work that another PhD student called named Sohini Deb. So Sohini actually did these experiments. She took over from where Deepanvita had stopped, her thesis finished. Sohini's thesis started, and one of the things that she was focusing on was how does ZOPQ protein suppress rice immune responses. Now, by this time, Mahesh Kumar Gupta, a postdoc in my lab, and Raj Kanwar together showed that ZOPQ protein has a phosphorimose hydrolase activity, but making specific mutations that we did in collaboration with Dr. Shankar Narayanan at CCMB. We made particular mutations that are expected to affect the biochemical activity of ZOPQ. We showed that they affect the biochemical activity of ZOPQ, but these mutants that are defective in biochemical activity were not involved in suppression of, so the mutants that are defective in biochemical activity can still suppress host innate immunity. So biochemical activity of ZOPQ is not needed for suppression of host innate immunity. So this is again something that Mahesh had done. So Sohini actually started looking at other sequence features of ZOPQ, and by that time there was other work done in other labs on other ZOPs of Xanthomonas, and also the Pseudomonas homologue of ZOPQ, which is called HOPQ, where they showed that these proteins, the ZOPQ-like proteins, bind to host 1433 proteins. So what Sohini did was that she made mutations in the 1433 binding motif of ZOPQ to find out whether these mutations would affect the ability of ZOPQ to suppress cell wall degrading enzyme induced immune responses in rice. So what she actually showed which I'll just show you in a slide, the data in the next slide. She showed that mutation of the 14-3-3 binding motif of ZOQ results in loss of the ability to suppress rice innate immunity. It turns out that ZOQ has two 14-3-3 binding motifs, and mutating one of them and not the other affects the ability of ZOQ to suppress innate immunity. And that's shown here. So if you infiltrate buffer or milky water into rice leaves, you get minimal callose deposits. If you infiltrate wild type BXO43, you again get minimal callose deposits. What Deepanvita did was she made mutations in those four ZOPs, ZOP N, Q, X, and Z, that are involved in suppression of cell wall damage induced innate immunity. And if all four are mutated in XO and you infiltrate that into rice, the quadruple mutant itself induces callose deposits, and we think it's because of the cell wall degrading enzymes that this mutant is still secreting. So the quadruple mutant was very helpful. This is a strain that Deepanvita developed. It took a lot of time to make 4 different mutations in one genetic background, but that mutant, when it was available, was very helpful because it itself is inducing innate immunity. Now into that, Sohini introduced the ZOQ protein on a vector called PHM1. So when you put the vector into the quadruple mutant, again the callose deposits are produced. So that means innate immunity of rice is induced. When you put ZOQ now into this quadruple mutant, that one protein is enough to be able to suppress host innate immunity. So ZOQ wild type, when introduced into the quadruple mutant, suppresses immune responses that are induced by the quadruple mutant. We also, in independent experiments, as I showed you, if you just introduce ZOQ, that will suppress cell wall damage, cell wall degrading enzyme induced immune responses. Now, this is a mutant that Sohini made in the 14-3-3 binding motif, where there was a tyrosine that she changed into an alanine, and this particular amino acid is thought to be a site for phosphorylation. So if you change it into an alanine, ZOQ now loses the ability to suppress rice immune responses. If you know, instead of alanine, if it is made into an aspartate, where it's a phosphomimic mutant, again it's able to suppress innate immunity. The second 14-3-3 binding mutant for ZOQ is around the T222 residue, and even if that is changed into
Alanine.
Yeah, alanine, basically there is no effect on ability to suppress innate immunity. Now, what this showed is that that mutation of one of the 14-3-3 binding motifs of ZOQ abolishes the ability of ZOQ to suppress rice innate immunity. And what Sohini went on to show is that rice has 8 14-3-3 protein encoding genes. That was already there from the rice genome studies. What Sohini showed is that ZOQ binds to 2 of these 14-3-3 proteins called GF14F and GF14G. And these proteins appear to be key signaling proteins in elaboration of rice immune responses. Okay, and it appears that ZOQ is able to suppress rice immune responses by interacting with these two 14-3-3 proteins. So this work actually Sohini completed, and it showed that ZOQ functions inside rice cells by binding to rice 14-3-3 proteins and suppressing host immune responses. So this part was done. Now I will get to the second part of my talk, where I will discuss about the function of ZOQ, the role that ZOQ has to promote virulence that occurring inside rice, inside XO cells. And I will show you data and try to convince you that ZOQ catalyzes the final step in cytokinin production, and that cytokinin is a key XO signaling molecule that controls bacterial transition from biofilm to planktonic state. So what Sohini was puzzled about was, if biochemical activity of ZOQ is not required for suppression of rice innate immunity, what is this biochemical activity doing? What is the role of the biochemical activity of ZOPQ in promoting virulence of XOO? Now, this is the reaction catalyzed by ZOPQ. So this particular compound, isopentenyl adenosine monophosphate, is the immediate precursor of isopentenyl adenine, which is an active cytokine. And a group in Switzerland had shown that the Pseudomonas homologue of ZOPQ appears to be involved in catalyzing the conversion of isopentenyl adenosine monophosphate to isopentenyl adenine. Their data were extremely convincing. So what Sohini did was, but they were not able to purify the protein. So what Sohini was able to do was purify ZOPQ and conduct these biochemical assays and demonstrate that indeed ZOPQ actually in vitro in a test tube converts isopentenyl adenosine monophosphate into isopentenyl adenine. So we knew now that clearly it is involved in, in vitro it can do this job of actually producing isopentenyl adenine. Now this is work that Gopalji and his postdoc did at NIPGR. They took this ZOPQ mutant and they showed that this ZOPQ mutant is defective in the production of isopentenyl adenine. And this is wild type XO. This is isopentenyl adenine in the supernatant. This is the amount of cytokinin that the wild type isopentenyl adenine that wild type produces. And this is the, the ZOPQ has a reduction in the amount of isopentenyl adenine it produces. Now, the reason it doesn't bring it down to zero is because we think that there's a second pathway for making cytokinin that XO has. And if you're interested, we can discuss that later. And into ZOPQ, if PHM1 plasmid is introduced, again, you have very low levels. But into the ZOPQ minus mutant, if you introduce the ZOPQ wild type protein, the levels of cytokinin are back to normal levels. But instead of ZOPQ wild type, if you introduce the biochemically defective mutants, so these were two mutants that Raj Kanwar and Mahesh had generated that convert, there's a residue at the 116th amino acid and a residue at the 279th amino acid, tyrosine at 279 and aspartate at 116, both converted into alanine, affect biochemical activity in the in vitro assays, and they also affect the ability of XO to produce isopentenyl adenine. So we knew now that yes, XO can make ZOPQ in vitro can synthesize the can catalyze the final step in cytokinin biosynthesis, and if we mutate ZOPQ in XO, it is reduced in producing isopentenyl adenine. So now what is the phenotype of the ZOPQ mutant? This is the wild type XOO. BXO43 is our wild type strain. And if you actually take up a culture supernatant and look at it under the light microscope, you can actually see cells that are mostly single, a few that are dividing. So you may get two cells that are attached to each other. What was surprising for Sohini is that these ZOPQ mutants are actually clumping together, and they were actually making biofilm-like structures, much bigger biofilms than the wild type cells. So this is actually what I'm showing you here, the clumps of the ZOPQ minus cells in comparison to what you find in the wild type. Because the wild type, the cells are in the supernatant and they're dispersed. In the ZOPQ minus mutant, they tend to be clumping together and stay towards the bottom of the tube. Now, if you add isopentenyl adenine to a ZOPQ minus mutant, the cells disperse from the aggregate state. So now we have a phenotype for ZOPQ in vitro cultures. So ZOPQ minus mutant forms these clumps and enhanced biofilms. And if isopentenyl adenine is added to the ZOPQ mutant, now they disperse. That's actually seen a little bit more pictorially evident here. These are BXO43 cells, and you can actually see this is done on fluorescently tagged BXO43, and these are BXO43 cells, wild type XO cells, and they're mostly as individual cells. But if you make the ZOPQ minus mutant, this red color indicates the level of fluorescence that is coming, and essentially it indicates clumping. So to this particular mutant, if you now add isopentenyl adenine, again the cells start dispersing. So ZOPQ mutants have this enhanced clumping or biofilm forming phenotype, which is rescued by adding isopentenyl adenine. Now, what was very interesting is that we are measuring cytokinin in the extracellular medium of wild type cells. So what Sohini thought was, okay, if I take wild type XOO, tag it with M-cherry, and ZOPQ minus mutant, tag it with EGFP, and you mix the two together, will wild cytokinin secreted by wild type complement the clumping phenotype that is observed in the ZOPQ minus mutant. So this is again work that we did in collaboration with Subhadeep and his PhD student. Biswajit and Subhadeep, we worked with Sohini. And so this is wild type BXO43. You can actually see individual cells. And this is tagged with M-cherry. And this is EGFP tagged ZOPQ. And you can actually see the clumping. When the two are mixed together, now you can actually see that even the EGFP tagged ZOPQ minus cells, they are actually now dispersing. So our hypothesis is that the cytokinin secreted by wild type now complements the deficiency of the ZOPQ minus mutant. And now the ZOPQ minus mutant also, instead of forming clumps, is actually making individual cells. Now, ZOPQ has a virulence deficiency phenotype. And this is the BXO43. And you can actually see the lesions here. The inoculation is done at the bottom of the leaves. And this is ZOPQ minus mutant. The lesion is actually somewhat limited. If cytokinin is added to the ZOPQ minus mutant, the lesions start extending and going further up in the leaf. So it looks like the virulence deficiency of ZOPQ minus mutant is primarily due to this effect on that is mediated by cytokinin. And we think that at some stage in the virulence process, XOO has to move from a plankton, a biofilm-like state. In the beginning, it has to attach. And then after a certain amount of growth, it has to become planktonic and move. And at that stage, this cytokinin helps. And ZOPQ minus mutant moves very slowly because it doesn't produce enough cytokinin. If you wait a little bit longer, these lesions will also start extending, but they never are as big as that of the wild type strain. So it looks like the virulence deficiency is because of lack of cytokinin production by ZOPQ. Now, okay, so this is ZOPQ, what ZOPQ is doing. Then we actually thought ZOPQ by itself cannot make isopentenyl adenine. Does XOO have the biosynthetic pathway for making isopentenyl adenine? It turns out that from primary metabolism, there is one enzyme that's called adenosine phosphate isopentenyl transferase. Just short form is IPT. IPT takes a compound called DMAPP. Again, you don't need to worry about what DMAPP is. DMAPP plus AMP will actually make this particular immediate precursor of, basically this is phosphoribosyl adenosine monophosphate, isopentenyl adenosine monophosphate. So now this is IPT acts to produce this, and on this ZOPQ acts to produce this. We knew that this activity is there, but we wanted to find out whether XOO has this second activity. And it turns out that IPT indeed has, XOO indeed has an IPT-like gene. And Sohini made mutants in this IPT gene. And again, this IPT mutant has the same phenotype as ZOPQ. So BXO43 has these single cells, or two cells, you can see them. But in the IPT mutant, you can actually see the clumping. And again, if you add isopentenyl adenine to the IPT mutant, you can actually see them dispersing. And this assay is again shown the same type of an assay I showed you for ZOPQ. You can actually see BXO43 here. These are the individual cells. This reddish orangish color actually indicates the clumping that is there in the IPT mutant. And if isopentanal adenine is added, they start dispersing. So we now know, and I'm not showing you the data here, but again, Gopal and his student, and also Chandan Kumar, who was actually at NIPGR at that time, they showed that even the IPT mutant actually now produces reduced amounts of exogenous cytokinin the same way as the ZOPQ mutant. Now, so far we know that yes, XOO can make cytokinin, and it has both the steps that are involved in actually making cytokinin, and if either one of them is blocked, cytokinin is not made, and you see this mutant phenotype. But so this only shows that XOO can make cytokinin, and it looks like it is secreting cytokinin. But the question then was, can XOO sense cytokinin? Is there a receptor for cytokinin in XOO? Now, this work had already been done a couple of years earlier. A group in China had actually shown that a relative of XOO called Xanthomonas campestris, Phytohormone campestris, which infects brassicaceous crops, it has a two-component system. This two-component system, they call them PCRK and PCRR, which is a sensor kinase and a response regulator pair. What they showed is that this two-component system senses exogenous isopentenyl adenine. And they proposed in their paper that XCC uses exogenous isopentenyl adenine. It senses its presence, it senses isopentenyl adenine, and induces expression of bacterial genes that are involved in adaptation to oxidative stress that's produced as part of the plant immune response. It turns out that XCC does not secrete isopentenyl adenine. It makes some isopentenyl adenine through a different pathway that I was telling you about, a second pathway. It doesn't have the IPT gene. Although it has ZOPQ, it doesn't have the IPT gene. And so it can't make this isopentenyl adenine. It makes limited amounts of isopentenyl adenine, and whatever is made stays inside the cell. And they propose that PCRK senses the extracellular cytokinin, and that initiates a signaling relay. The response regulator gets activated upon PCRK binding to cytokinin, and then a downstream signaling transduction cascade starts. And Prabhu Patil at IMTech has done a lot of work to show that IPT gene is absent from XCC, and it looks like IPT is introduced into XOO by horizontal gene transfer from another soil bacterium. So this is work that I will not be talking in detail, but Prabhu and his student have done that work. So now Sohini went back to look at the XOO genome. It has PCRK, PCRR two-component system, and Sohini made XOO mutants that are defective in both of them. Now, the XOO, PCRK, and PCRR mutants exhibit the same cell aggregate enhanced biofilm phenotype as the ZOPQ and IPT mutants. But addition of exogenous cytokinin to cultures of PCRK and PCRR does not rescue those phenotypes. And that data I'm showing here. So just I'm showing you for PCRK, not showing you for PCRR, but same data. So this is BXO43, and you can see the individual cells here. PCRK, the cells start clumping. You can actually see this big clump. But if you add isopentenyl adenine, those clumps don't disperse. We think that is happening because even if you add exogenous cytokinin, because the receptor is not there, XOO cannot sense the presence of cytokinin. So even if you add exogenous cytokinin, it doesn't actually, the phenotype is not rescued. And this is the biofilm phenotype. So this is BXO43. The purple coloration indicates the extent of biofilm. This is PCRK minus. You can actually see enhanced purple and enhanced, that means enhanced biofilm. Even if you add isopentenyl adenine, that doesn't change. Similar phenotype for PCRR. Wild type has a certain biofilm phenotype. this makes more biofilms, and adding isopentenyladenine doesn't change. So our model currently is that XOO uses IPT and ZOPQ to make isopentenyladenine, and this isopentenyladenine is secreted into the medium, and this IP that is in the extracellular space is recognized by PCRK and PCRR to regulate genes that are involved in the biofilm to planktonic transition. So a model in terms of the dual functions of ZOPQ are that IPT and ZOPQ are involved in actually converting DMA, PP, and AMP into isopentenyladenine. This gets secreted out of the cell, and this release of isopentenyladenine switches XOO from biofilm to planktonic state. So that is one phenotype that's within the bacterial cell. And this ZOPQ also has a second function where it gets into XOO and it binds to Rise 14-3 proteins and suppresses immune responses. Now, we don't know whether ZOPQ has an additional function. ZOPQ is also involved in making a cytokinin inside rice cells. We know it makes cytokinin inside XOO cells. We don't know whether it also is involved in making cytokinin inside rice cells. But it is possible that it could be doing it. So what I hope I've told you is that we have shown very clear evidence that cytokinin is a signaling molecule in XOO. Now, once we found that, we started looking at the literature, and we found that several other people had reported that bacteria make cytokinin, including Mycobacterium tuberculosis. They have shown that Mycobacterium tuberculosis produces cytokinin, and there are other bacteria that encode predicted cytokinin biosynthetic functions. But it was not, these are bacteria that don't associate with plants, that are not known to associate with plants. So either they have an association with plants for which they have to make cytokinin that we don't know about, or cytokinin has another role in bacterial physiology that is independent of what it does in the plant. So what we think from our data in XOO is that we think that cytokinin may be, our data suggests that cytokinin is a signaling molecule in XOO, and we think that many other bacteria may actually use cytokinin as a signaling molecule, because many bacteria have functions that are required that are involved in making cytokinin. And we think that it is possible that cytokinin actually evolved as a signaling molecule in bacteria, and that plants have subsequently taken the same functions, enzymatic functions. And if you, those who may remember who work on cytokinins, know that the plant receptor is also, for cytokinin, is also it's a two-component regulatory system that's involved even in plants. So we think that the machinery that was there in bacteria is taken by plants and much more elaborated to control many, many more functions within plants. So that's what we think is happening. But that's a hypothesis we have only shown in XOO and not done in any other bacteria. So these are acknowledgements. As I said, a lot of the work is Sohini Dev, and Hiten has been involved in guiding Sohini and giving inputs in terms of the work, not only for Sohini, but to Mahesh, Raj Kanwar, and others. So Deepanvita, Gokulam, Chandan, Palash have actually worked on this, and Chandan worked also at NIPGR. He came with me from CCMB to NIPGR, was involved in several projects, and towards the end he started working on this particular project and continued with Gopalji. Subhadeep and Biswajit Samal at CDFD, and Prabhu Patil and Amandeep Kaur. So thank you all for your attention. Yeah, I'd be happy to take any questions.
So Ramesh,
Very nice.
Yeah, sorry.
A very nice work. So I was just wondering, so this ZOPQ have two different function in two different organisms. So you have already mapped that which domain interacts with 14-3 in the plant. So is it the same domain or some other domain which interacts when it is converting this IPMP to IP in bacteria? So
Okay. Yeah. No, the biochemical activity is not in the in that near that S65. The serine 65 is not involved in the biochemical in the biochemical activity. So that's a different domain.
Okay. Okay. Thank you.
Thank you.
Ramesh, I have a quick question, and also like a thought maybe. So since this is an cell signaling kind of molecule, the cytokine derived molecule. So do you think the concentration which is required required to have the threshold to activate the bacterial signaling system is a little different from what concentration which it encounters in the plant? Otherwise, in planta they could have it would have been complemented without supplementation of exogenous cytokine.
Yeah, that's a good question, Subhadeep. Long and the short, I don't have the answer for it because we don't know how much cytokinin is made by rice cells which are infected with XOO and which are not infected with XOO. But the fact that the ZOQ minus mutant is virulence deficient just to us that whatever cytokinin it can get from the plant is not enough for that switch.
Not enough, yeah.
It has to supplement its own synthesis.
So the threshold concentrations for bacterial signaling is quite different from what the threshold concentration which are required for influencing.
I don't know about that because see, XOO grows within the xylem.
Okay. Yeah.
How much function cytokinin has within the xylem, I'm not very sure. So cytokinin concentration within the cell of rice, that may be more. And cytokinin within the xylem may be much lesser. Okay, so that
Ramesh, is also possible that cytokine which is made by the Xanthomonas itself, the local concentration will be high intracellular as well as in the periplasm, so that it activates the.
Right. Correct.
Yeah.
That's the idea. But we have to actually quantitate within the plant. Mostly focusing on the in vitro culture. Look at what happens within the plant. Yes.
Okay. Thanks.
Hey, is time there? Can I ask questions?
Please.
Ramesh, that's a great talk. It's excellent. So actually, I was thinking, like, if you look at agrobacterium, that also injects IPT gene.
Right.
And just the previous step of the cytokine biosynthesis to actually facilitate the growth.
Correct.
And here also is a type 3 secretion effector injecting cytokinin biosynthetic gene enzyme in the cell. So do you think that this is also towards increasing the hypertrophy or any kind of cell division to induce metabolism, maybe to increase? Is it possible?
We don't have evidence for that yet. Okay.
Suppose if you take a culture filtrate of this Xanthomonas and if you apply on a cell, does it induce cell division? Is it a biologically active cytokinin for the plant?
What we have to do is, okay, we have not again checked what will happen because the culture supernatant, we have not taken the culture supernatant of XOO and seen whether that will induce cell division in callus cultures. Yeah, that's a good point. We can try that, but we have not tried that. A little concern there has been whether the spent medium after bacterial growth may be having, it'll have multiple things, whether it'll have things that will inhibit the callousing, but only when we do it, we will know.
Sir, maybe you can take cell wall degrading mutant, type 2 mutant, and then.
Okay, no, but even then, just growing up a culture of the bacterium, that itself, there'll be waste products of bacterial metabolism that will accumulate in the culture. So we have to separate out the cytokinin that's made by the bacterium. And once we do that, then, what are the concentration, all those become an issue. So, but it's a good. We can give it a try. We can give it a try.
One question actually, because I do not know. So you said it's growing in xylem, that I know, but does it actually also deliver the type 3 effectors in the xylem cells, or most likely not, right?
We think it delivers them into the xylem parenchyma.
Xylem parenchyma cells.
Yeah, xylem parenchyma it introduces. Now, whether it will go beyond and introduce into other cells, Subhadeep and Biswajit have shown that XCC can actually break out of the xylem and get into other things, other tissues. Well, XOO, I'm not sure whether that actually happens. But if that happens, then it can actually start introducing it into palisade parenchyma cells also. But definitely xylem parenchyma it does, because it must introduce into living cells to be able to get the nutrients.
That is what, yeah, right.
Yeah,
okay. Okay, thanks, Ramesh.
Sure, thank you.
Sir, there are two more questions.
Yeah, please.
In the chat box.
Okay, shall I look at them?
One is straight, how does plant differentiate between bacterial cytokine and self-cytokine?
Yeah, actually, yeah, that's again, I think many people are asking in the similar way. It's a very good question. We actually don't think there's a difference between the two. The isopentenyl adenine that the bacteria makes and the isopentenyl adenine that the plant make, we don't think really that there is a difference between the two. Okay, so maybe there are concentration differences that occur. We have to actually do very detailed experiments to see how much cytokinin is made within rice xylem fluid, how much is made after XOO is grown in those xylem fluids. So we think it's the same. It is the same molecule, but whether there are levels, the levels of cytokinin may change after bacterial infection, and how that may influence plant physiology. That is what Ashish was also asking. We really don't know all of that.
The second question is, cytokinin is found to be produced in root as well. So would the phenotype of XO cell clumping will be the same as seen in leaves of rice?
Okay. No, one more point I have to clarify here is that we are looking at clumping only in the. We have not looked at the clumping phenotype within the plant. We are only looking at clumping phenotype within the test tubes, and we think that it is helping within the plant. That same thing is happening within the plant. Again, it is possible to do these experiments. We have not yet done those. But if you ask me, unless the cytokinin that's present in the xylem of the leaf is also contributing to the phenotype, I would expect the same thing to happen whether it is in the root or in the leaf.
Sir, Pranam, and kindly keep your blessings on us. We'll go to we have a panel discussion again after this session. We all will again have discussion on such an elegant, we are learning. So next is our Dr. Gopal will be speaking. Gopal is from NIPAM, we all know. So Dr. Gopal, you please start your talk.
Can you see my slides?
Yes.
Thank you. So it's a pleasure to get this opportunity to give my presentation, and I'm thankful to Subhadeep, Subhendra, and all the organizers to give me this opportunity. And particularly, it is really exciting to talk just after Ramesh sir. So I think I will come up to his expectation. So the title of my presentation is Conversion of a Defensive Toxin Antitoxin System into an Offensive T6 SS Effector in Burkholderia. So a couple of years back in my previous MIPAM presentation, I have been discussing how we identified a very interesting bacterium from rice. This bacterium is Burkholderia gladula strain NGJ1. It is yellow in color. Though Xanthomonas also looks yellow, but this is different yellow than Xanthomonas. And this is the fungal pathogen, Rhizoctonia solanae, which causes sheath blight disease. This looks under laboratory media like this. And what we observed that this bacterium has an unusual property to feed on Rhizoctonia solani. That means when these two are allowed together to grow, we see the bacterium is growing over the entire fungal biomass and eventually eat away the fungal biomass. So this property is called mycophagy, fungal eating property, and we have been trying to characterize the fungal eating property of this bacterium. And I've discussed this in my previous slide that the bacterium uses type 3 secretion system to deliver a prophase tail-like protein to feed on fungi. And this work was published in Nature Communication. So in today's talk, I'm going to talk to you about another interesting property of this bacterium, which is antibacterial property. So what we observed that besides eating fungi, this bacterium has also a strong broad spectrum antibacterial activity. So this is the bacterium. And you can, for example, on this plate, the E. coli is spreaded, and we put this Burkholderia at 3 different places, and you can see the zone of inhibition. The zone of inhibition reflects antibacterial activity on E. coli, similarly on Agrobacterium, and a strong zone of inhibition on XO. So I'm just keeping 3 examples, but I have tested around 20-30 price-associated bacteria. And all of them, this bacterium, almost all of them, except 2 or 3, this bacterium demonstrated antibacterial activity. So we started exploring what is the basis of antibacterial activity of this bacteria, NGJ1. So there are reports that bacteria uses type 6 secretion system. This is a little different than type 3 secretion system, but also it's like ejectosome. So the mechanism is broadly similar, that using this type 6 secretion system, and the bacterium injects protein, effector protein from cytoplasm into the directly into the eukaryotic cells. And this bacterium is also shown, this secretion system is also shown to be used by bacteria to kill other bacteria. So the T6SS has dual functions. The bacteria uses T6SS to deliver protein directly into eukaryotic cell, and it kills the eukaryotic host to establish infection. At the same time, bacterium uses T6SS to target cohabiting bacteria to kill them. So what we observed that NGJ1 encodes 2 different T6SS apparatus. It's not required to remember the name of the genes. You just take it from me that there are 2 different T6SS apparatus, and both of them are complete. So basically, XO can make 2 disinjectosome-like apparatus during colonization or during bacterial interaction. So we have started exploring what is the function of this T6SS effector with respect to the antibacterial activity. So we disrupted both this T6SS1 and T6SS2 individually and observed that disruption of either T6SS1 or T6SS2 compromises the NGJ1 ability to kill bacteria. So this is the control bacteria. You can see E. coli is growing up to some level. However, NGJ1 is preventing the growth of E. coli cell. But when we disrupted either T6SS1 or T6SS2, the bacterium was compromising antibacterial activity. And similar is true for Agrobacterium. So this demonstrates that XOO requires both T6SS1 and T6SS2 for antibacterial activity. That means either you disrupt T6SS1 or T6SS2, bacterium is deficient in, at least deficient in antibacterial activity. So we have started exploring how these T6SS effectors contribute to the antibacterial activity. So we have the genome sequence of NGJ1 available. So we computationally mine what all are different T6SS effectors. And these T6SS effectors are very interesting organization. Ideally, they are encoded in a operonic fashion. And I'm showing you few of the operons of T6SS apparatus. So the take-home message is that for each effector protein, there is an immunity protein, because these effector proteins are antibacterial. So the bacteria has to protect itself from its toxin activity. That's why for each T6SS effector, there is an immunity protein, and that is supposed to prevent effector-mediated intoxication, self-intoxication. At the same time, in the host or in the cohabiting bacteria, only effector is secreted, immunity is not secreted. So the effector will be delivered to kill the cohabiting bacteria and sometimes into the host cells. So using this operoning structure, we identified 14 different T6SS effectors of NGJ1. another interesting feature of T6SS effector is operon is that beside the effector and the immunity protein, there are certain protein, accessory protein that is required for T6SS mediated delivery of this effector. So you don't remember name of these things, but take it that either they have either VGRG or PAR, which enables this effector protein to be secreted into the host cell. So what we started testing is whether these putative computationally predicted T6SS effector immunity function is effective. So for that, we clone this effector and the immunity protein and heterologously express in E. coli cells. So here I am showing you that if without IPTG, so it is under clone under IPTG inducible promoter. So when one particular effector 9 TLE is cloned and expressed in E. coli without IPTG, the E. coli cell is growing happily. But when we put IPTG, that means it will lead to the expression of the effector protein. So the E. coli cells started killing. So this shows that the expression of the effector proteins kill the E. coli cell. However, when we try to co-express the immunity protein using another plasmid, so now co-expression of the immunity protein is protecting it. So that this is mostly observed for most of the T6SS effectors, which we have predicted from NGJ1. So that means ectopic expression of the effector protein kills the E. coli cell, while co-expression of the immunity protein protects it. So this was for majority of the effectors, but interestingly, what we observed a novel kind of T6SS effector, which we have named it TSC-TBG. I will describe this in detail in my next rest of the talk. So this effector protein, when we induce and when we express effector TSC-TBG effector protein, the E. coli is significantly killed. So it is very potent antibacterial protein. However, the co-expression of the immunity protein, which was just encoded next to the TSC-TBG effector protein, the immunity protein is not protecting it. So this is very interesting. How come this bacteria encodes a toxic protein? And that is antibacterial, but at the same time, it's not the immunity protein is not effective against it. So what we started trying that how it is happening and what is so special about this immunity protein. So what we observed is that when we express as a transcriptionally fused protein, that means earlier we were expressing the effector on one plasmid and immunity on one plasmid and trying to see whether it is protecting. But when we transcriptionally fuse both the effector and protein under the same promoter, then it started protecting it. So, for example, this case, the transcriptionally fused effector immunity protein is providing protection. So, and further we observed that the immunity protein contains an IMP52 domain. So, the TSC-TBG proteins contain IMP52 domain. And if you disrupt this IMP52 domain, then the disrupted mutant variant of this TSI-BG protein has to protect it. So we concluded that the stoichiometric ratio of the effector and the immunity protein is important for the protection. So for the rest of the effector proteins, stoichiometry was not important. When you co-express them on two different plasmids, it was protecting. But this particular effector protein, stoichiometry is important, and only if they are expressed together in an equal stoichiometric ratio, it is protecting it. So we observed that NGJ1 encodes 3 paralogs of this kind of effector protein. We named it TSC-TBG1, 2, and 3. So all the operonic structures are same. That for effector protein, there is the immunity protein. Each of the effector protein has the immunity protein. And also there are conserved accessories protein, which is required for T6SS mediated delivery. So interestingly, what we observed that each of these TSC-BG effector protein contains TOX-REH5 domain. So TOX-REH5 domain is encoded in each of the effector proteins. And what we observed that this TOX-REH5 domain shares a structural similarity with endonucleases. And there are certain aspartate and lysine residues that are conserved in the active sites of each of these effector protein and also the endonucleases, which is to degrade the nucleases. So this suggested that maybe this TSC-BG protein also function as endonucleases to prove it, indeed, whether it has endonuclease activity, we purify these effector proteins and observe that it effectively degrades the E. coli DNA, but it fails to degrade the NGJ1 DNA. So here I am showing you that we purify both two different TSCBG, 1 and 2 effector proteins, and treatment with the purified proteins degrade the E. coli genomic DNA. And in absence of the protein, you can see the band. And also when we disrupted this active site residue, we converted into aniline. We converted aspartate and lysine residue into the aniline. And the disruption, the changes in the active sites make this protein defective. So the active site variant is unable to degrade the E. coli cells, E. coli genomic DNA, but at the same time, both the TSCBG protein is effectively degrading the E. coli genomic DNA. But interestingly, we observe that the effector protein is not able to degrade the genomic DNA of NGJ1 own. So it fails to degrade its own genomic DNA, but at the same time, it is degrading the genomic DNA of the E. coli cells. Further, we observe that it is not just degrading the genomic DNA, it is degrading the plasmids and anything from the prey bacterium. So, for example, there is a PHM1 plasmid which has broad host range and which can replicate in different bacteria. So, the PHM1 plasmid, when isolated from NGJ1, was not getting degraded. You can see the PHM1 plasmid from NGJ1 is not able to be degraded by this TSCBG protein, while PHM1 from E. coli cell was getting degraded. So we thought that maybe there is some host specificity, and like restriction endonucleases, there is something, the host nuclear DNA is different, and there may be some methylation or something which is protecting the host DNA from this nuclease activity. And indeed, we identified a DNA methylaminase DAM protein, which is encoded next to this effector operon. So next to this immunity protein, after one gene, there is one DAM protein encoded. And that DAM protein, when we ectopically express now in E. coli cell, that now that plasmid is isolated from DAM plus heterologously expressing DAM in E. coli cell was not degraded, but without DAM, the plasmid was degraded. So this confirms that this TSCBG protein exhibits host DNA methylation sensitive DNA activity. So this is an interesting activity of the T6SS effector protein, which people have not described before. What we observed that beside DNA activity, the TSCBG protein also have potent RNA activity. Earlier, I told you that DNA activity is only on the prey bacteria, but it was not observed on the native whole self bacteria. But RNA activity is pertinent to both self and the prey bacteria. So here I am showing you that RNA activity is there for the NGJ1 RNA and as well as the E. coli RNA. And also the disruption of the active site residue is not able to kill the RNA activity. So that shows that RNA activity is differently regulated, differently governed than the DNA activity. The maybe active sites are quite different. So NGJ1, this is the NGJ1 RNA. When upon treatment with TSCTBG, effector protein is totally degraded. Disruption of the active sites does not disrupt the RNA activity. We observe the RNA activity even after disruption of the active sites, and similar is observed in the NGJ1 E. coli RNA as well. So we then was very intrigued to know how the RNA activity is degraded. I told you the DNA activity is protected by DAM, DNA which is encoded in NGJ1. So NGJ1 is this effector nuclease activity is not observed on the native DNA, but RNA activity, how the NGJ1 is protecting. So whether the immunity protein protect NGJ1 from the RNA activity of the TSC effector protein was the next question. So what we observed that this immunity protein, which was associated with the effector protein, contains, I told you that it has IM52 domain, which was required for effector neutralization. What we observed that beside IM52 domain, it also harbor a helix-turned helix domain, though it is not very orthodox helix-turned helix domain, it is non-canonical helix-turned helix domain, but there is a helix-turned helix domain at the end terminus of each of these TSCTBG protein, the immunity protein. So this suggested helix and helix domain suggested that the immunity protein may have transcriptional regulation activity. So we tested whether now this immunity protein also exhibits transcriptional repression activity and thereby regulates the expression of this operon and protects it from the self-degradation. So to prove that, we analyzed the promoter sequences, and what we observed, there are two promoters in this operon, effector operon. I told you that effector protein, immunity protein is encoded along with some accessory protein, and in bacteria they are organized in operonic fashion. So we observed one promoter upstream to this part protein. So we call it P external protein, a promoter. And if this promoter is activated, now the entire operon will be transcribed and we get the expression of each of these. However, we observed there is an internal promoter also, and which we have named it P internal. And if this P internal promoter is active, only the effector and immunity protein will be transcribed and there will be no effector protein. So this will be very interesting scenario. And when external promoter is activated, entire operon is transcribed, and so there will be expression of the SH-rich protein, which will enable T6SS mediated secretion. But if the internal promoter is active, only effector and the immunity protein will be transcribed, and that will only have the intracellular function, and also it can be that can have intracellular function. So to test this hypothesis, we first started exploring whether both the promoters are active in our bacterium or not. So we did promoter gas reporter assay, and we cloned each of these promoters in front of the gas and observed that both the promoters are active in our, can drive the expression of the gas gene. So that shows that the both promoters are active. And further, what we observed, that when we ectopically express the immunity protein, now the gas expression is repressed. So for example, in absence of IPTG, we could get the gas expression. However, in presence of IPTG, when this immunity protein was expressed, the gas expression was suppressed. So that shows that immunity protein represses the expression of the gas gene. And I told you the immunity protein have the IM50 domain and helix and helix domain. So what we observed that when we disrupted the IM52 domain, which was required for effector neutralization, the transcriptional repression activity was still retained. So in absence of the IM52 domain, the transcriptional repression activity was still retained. However, when we disrupted the helix and helix domain, the repression activity is gone. So this shows that the expression of the immunity protein is repressing the expression of the promoter, both internal promoter as well as the external promoter. And we saw that the helix and helix domain is required for the transcriptional repression activity. To prove this further, we did ELMSA assay and showed that indeed the TSI immunity protein has ability to bind to the promoter of this external promoter as well as the internal promoter and leads to the shift in the DNA. And however, when we disrupted helix and helix domain, there was no shift. So this suggested that indeed the immunity protein binds to the promoter and regulates represses the expression of the promoter. So this was shown in case of the external promoter also as well as the internal promoter. And in both the cases, helix and helix domain is required for promoter binding. So I told you that the effector protein exhibits RNase and DNase activity. There was interesting observation that DNase activity was only specific to the prey bacteria, while RNase activity was observed on both prey bacteria as well as the cell bacteria. And I told you that the TSI immunity protein, which was associated with the effector protein, neutralizes the effector proteins either through direct interaction, but there then stoichiometry was important. You have to express them in a proper stoichiometric ratio, and also they regulate the effector operon through transcriptional repression activity. So this is very interesting, and take it from me, this kinds of interaction was not shown in any known T6SS effectors, but it has the similarity, the functional analogy we could make with toxin-antitoxin system in bacteria. So toxin-antitoxin bacteria encodes toxin-antitoxin system for its intracellular functions, and they are known to help the bacterium for stress adaptation. So here only toxin and antitoxins are encoded and they are not secreted. So there are no SS-rich protein, and under a certain stress condition, the toxin, antitoxin proteins are degraded, only toxins are produced, and that makes bacteria to undergo self-arrest and things like that. However, in case of under normal condition, immunity protein is there, which will antagonize the effector protein and also transcriptionally represses the expression and keep this toxic protein inert. But under a stress condition, the immunity protein is degraded and now the effector protein can exert the intracellular function. So what we observed that I told you there was two operon promoter, there was two promoter sequences, external promoter sequences, and when external promoter was active, all these operons was, all these genes were transcribed. So in that case, we considered it as T6SS operon because this PAR protein will enable the secretion of this effector protein using type 6 secretion system. However, when internal promoter was active, only effector and the immunity protein will be expressed, and in that case, we started considering it as toxin-antitoxin system. So now we started exploring whether these kinds of domain structures are present in other bacteria or not. So what we observed that these effector proteins and the immunity proteins are widely conserved in various bacteria, but they are encoded in either one of the two operonic structures. So in many of the bacteria, they are encoded as toxin-antitoxin system. For example, in actinobacteria, most of them are encodes these proteins as toxin-antitoxin system. That means there is no associated protein which will enable it for secretion. However, in gamma proteobacteria and some of the beta proteobacteria, they are encoded as both toxin and as well as TH system as well as the T6SS effectors. So this was interesting that bacteria is encoding either them T6SS effector or TA effector. So to have more insights, we started analyzing the different Burkholderia genome to see whether they are encoded as TA or T6SS effectors. So in many Burkholderia, we observed that they are encoded either as toxin-antitoxin system, and in other Burkholderia, we observed them as encoded as T6SS effector. And I told you that there are SS-rich protein encoded. That's why we started considering them T6SS effectors. So what we observed that there are certain transposable elements, insertion sequences elements present at the same locus. So we observed certain Burkholderia where the effector and the immunity protein. So for example, in this pseudomoli, Burkholderia pseudomoli, this effector and the immunity protein is encoded alone. There is no T6SS effector. But there is another strain of pseudomoli. There are part of the SP protein that enable us to do T6SS effector. But interestingly, we identified another pseudomoli strains wherein there are some transposable elements. So these blue boxes, IS3 elements, for example, in this pseudomoli, you can see the IS3 elements, two IS3 elements encoding flanking to the DUP and the PAR protein. However, in another pseudomoli strain, we observe one of these IS3 element is deleted and only one IS3 element is present. And in pseudomoli, another pseudomoli strain at the same genomic locus, now both the IS3 element is disrupted. So this helped us to anticipate that maybe the IS3 element is trying to enable the bacterium to adopt this T6SS accessory proteins and has helped the bacterium to convert an intracellular toxin as an extracellular weapon to kill the cohabiting bacteria. So to emphasize this more, we think that bacteria encoded toxin-antitoxin system as a canonical toxin-antitoxin system. There is no accessories protein to enable it to secrete it into the host prey bacteria. However, the IS3 element, one particular IS3 element has brought this DUP gene and the PAR gene and inserted it within the toxin-antitoxin operon. And due to this, there is an operonic fusion of the toxin-antitoxin gene along with the PAR and the DUP gene. And also during operonic fusion, there is a creation of a new ORF that is called hypothetical protein, and Nature microbiology paper has demonstrated this hypothetical protein acts as a co-chaperone, which is essential for secretion of this toxin antitoxin protein into the host bacteria. So we empirically demonstrated that a transposable element has brought T6SS protein, fused it into the toxin antitoxin operon, and there is an operonic creation due to the transposition and excision during excision. As we know that transposition during excisions do some duplication in the sequences, and due to that duplication of the sequences, one hypothetical ORF has been created, and now the PAR, DUF, and the hypothetical ORF has been fused along with the toxin and antitoxin system, which has converted this intracellular toxin into an extracellular weapon. So what we think that bacteria generally under normal condition, that immunity protein transcriptionally represses it, and there is no expression of the effector immunity protein at all. But under a stress condition, the immunity protein is known to be thermolabile, so an unstable protein, so it is degraded, and now the transcriptional repression is over, and now the toxins are released, and now these toxins are delivered into the into the prey bacterium using this accessory protein PAR, DUF, and the hypothetical protein, and secreted into the prey bacteria. So during intracellular function, these toxins can degrade the RNA and can enable the bacteria to undergo growth arrest. And this is now shown in other mycobacterium and another bacterium, that bacteria can form persister cell formation and can undergo, let's say, a stress adaptation response. I can repress the growth to adapt to the stress condition. And here we saw that the bacteria has converted known to secrete this effector protein using type 6 secretion system into the prey bacterium. And now in prey bacterium, it can either degrade the prey bacterial DNA and also can degrade the RNA. And with dual nuclease activity, it kills the prey bacterium. So we have empirically demonstrated that there is a conversion of a defensive toxin antitoxin system into an offensive T6SS effector in Bucholderia. So this work has been published this year in EMBO report, and I must acknowledge the hard work of my students. So I told you that this bacterium has an intrinsic ability to modulate the dynamics of the plant microbiome. And as you all know, that microbiome has profound impact on the growth and health of the plants. And we emphasize that the microbe-microbes interaction can play a crucial role in shaping the dynamics of microbiome. We have exemplified how a rice-associated bacteria and other related bacteria had converted its intracellular defensive toxin antitoxin system into an offensive extracellular weapon to kill the cohabiting bacteria. So to give you an overall picture of how this Bucholderia and plant microbes interaction is shaping the dynamics, what we feel that NGJ1 through its antifungal and mycophagous activity is trying to utilize the endophytic fungi as a nutrient source. However, when these nutrients are released into the extracellular milieu apoplast, it is a source of nutrition for other cohabiting bacteria. So to control the competition, to win the interbacterial competition, that now NGJ1 has learned to use type 6 secretion system to kill the cohabiting microbes and prevent the competition and be a hub bacteria, hub microbes, which can shape the dynamics of the plant microbiome. So we are now working towards understanding how a bacterium can shape the microbiome dynamics during plant colonization and what is the implication in plant health and plant protection. So I must acknowledge the hard work of my students. So most of this antibacterial work has been done by single-handedly by Sunil Yadav, who has finished his PhD, and he really has been a great student who could push the entire work from his own. And the mycophagy work has been done by Sunil, Durga Madhav Swain, and rest of the lab members has been contributing extensively. And I must acknowledge Professor Ramesh Sonti, my mentor, my supervisor, who has been a guiding force throughout. And he had been a great help for me to draft the manuscript, plan the experiments, and also shape them into the format so that it's easier to get it published. So I'm really thankful to him, and I'm gratefully to all funding agencies. NIPGR has been a great support for me. The present and past directors has been really a fabulous support for me, and it gives us funds. Also, I've been generously funded by DBT and recently Swan Jayanti Fellowship at CRB. So thank you.
Nandi sir, you want to you can ask some question.
Okay, such a great work, Gopalji. It's so good. Some small just curiosity, like say, if you look into the methylation level in the bacteria is always very high, at least compared to the eukaryotes. So this DNAs, which probably still active, more active when you have more reduced methylation, right? So.
Yes. So actually what we feel is host methylation sensitive. So it is somehow discriminating the methylation pattern, let's say in prey bacteria, and methylation pattern in the cell bacteria, native bacteria. So we feel the DNA dam of NGJ1 is making some host methylation specificity, which is enabling this the effector protein to be less prone to degrade. So now we are trying
overall, if you look at the methylation frequency difference between E. coli and Bacteria, do you anticipate it will be highly different?
Not sure actually. It is not that maybe different, but there are two kinds of methylation: adenine methylation and cytosine methylation. Right. So I think cytosine methylation is mostly conserved, but adenine methylation is specificity is there. So we are trying now to unravel the what we nicely observed, that PHM1 from NGJ1 and PHM1 from E. coli is differentially degraded. So we are same plasmid is there. So in general, the methylation pattern should be the same. So now we are trying to isolate this plasmid from NGJ1 and plasmid from E. coli and trying to map the methylation status of this plasmid to see whether there is any specificity. So we are on the job actually. So right now
you did not try digesting fungus DNA or rice DNA?
That rice and fungus DNA is degraded by both actually.
It's degraded.
Yes, so only native DNA is not degraded, rice and fungal DNA is degraded.
Okay, great work. Thanks, Gopalji.
Thank you.
Congratulations.
Sir will ask question. Sir.
Gopal, nice work and nice presentation. See, I have one query. You mentioned in the beginning of the talk that there are two type 6 secretion systems.
Yes, sir.
So are some of them, are one type 6 secretion system required for killing certain bacteria and other for some other bacteria? Why does NGJ1 require two type 6 secretion systems? Sure. Is there any specialization in terms of the prey bacterium or in terms of the effectors that are secreted through one and not through the other?
Sure. So we have been asking this question. So as we observed that there are 14 different T6SS effectors in our bacterium. So we feel that some of them is targeted through type 6-1, T6SS1, and others through T6SS2. That's why disruption of, but there are some which can be secreted by both. So what mechanism, what we are seeing is there are accessory protein, for example, this BGRG and PAR. So what we feel that in T6SS1, there is BGRG. In T6SS2, there is both, there is no BGRG. So what we feel that BGRG containing effectors are secreted through PAR containing apparatus, and that PAR containing effectors are secreted through BGRG containing apparatus. So that is our hypothesis. We are now trying to see how this T6SS1 and T6SS2 are different, whether they are differentially regulated in presence of the host bacteria, means prey bacteria. So there is SCP protein. We have antibody for, there is one SCP protein on T6SS1 apparatus, another SCP protein on the T6SS2 apparatus. We have antibody for both of them now. We are trying to see whether particular bacteria can only induce SCP1 or another bacteria is inducing SCP2, or both are induced in presence of certain bacteria. So we are now going into that direction, sir.
Okay. Thanks.
And another interesting thing what we are observing is, that is yet not published, so that there is some effect of intracellular trafficking in the prey bacteria. So imagine these effectors are secreted into bacterial cytoplasm, but there are different functions of these effectors. Some are supposed to be periplasmic, some are supposed to be working in the nucleus, some are working to affect the cell wall components of it. So ideally, they should be targeted. So what we have recently observed is that if you disrupt the subcellular localization signal of this effector protein, it is modulating its antibacterial activity. And what we know that in bacteria, there are Sec-dependent secretion and the Tet-dependent secretion. And we have procured the Sec and Tet mutant of E. coli. And interestingly, the Sec mutant is not getting degraded, killed by the NGJ1 cell. And the Tet mutant is getting killed. So that is a clear demonstration that intracellular trafficking is important for effector function. So that may be another interesting work which we are developing in the lab.
Yeah, that's very interesting, Gopal. Sure. Very good. Very nice.
Thank you.
It's very beautiful, Dr. Gopal. Very nice. So again, we'll have the panel discussion at the end. When now we'll be listening to Dr. Divya Chandran from RCV. Doctor.
Yeah, good
Okay.
morning, everyone.
Namaskar, namaskar.
Namaskar. I'm just going to share my screen. Okay, so please let me know if you can see my screen, right?
Yes,
okay.
yes.
So, good morning, everyone. It's an honor to be a part of this group, and thank you for inviting me to be a part of this group. And thank you to all the organizers of MIPAM 2022, Shubhadeep, Dr. Darren Thomas, Dr. Ray, everyone, for giving me this opportunity to present my work here. So in our research program, we study the molecular interplay between a fungal pathogen, which is commonly known as powdery mildew, and its host leguminous plants, with the long-term goal of engineering durable resistance against this disease in legumes of agronomic import. And today I'm going to discuss some of the new insights that we have gleaned from the molecular mechanisms underlying this host pathogen interaction, particularly on the role of the isoflavonoid phytoalexin pathway in powdery mildew resistance in Medicago truncatula. So before I go on to the actual study, I'd like to briefly introduce the pathosystem that we work on, which is the powdery mildew Erysipecae. So Erysipecae is a fungal pathogen that causes significant yield losses in both crop and forage legumes, for example, pea, lentil, fava bean, to name a few, and forage legumes, just Medicago truncatula, alfalfa, and vetch. And this fungus infects only the aerial parts of the plant, and it produces this white powder-like symptoms on the surfaces of the leaves, stems, and in some cases even on the fruit. An important feature of this fungus is that it's a true obligate biotroph, which means that it can only infect and survive on living host plants. And this makes it extremely challenging to work with. Now here I'm showing you just a microscopic view of a single Erysipecae colony on the surface of a pea leaf. And as you can see, majority of the fungal structure is present outside the plant cell on the surface of the leaf. And the only infection structure that's present within the leaf, that too outside the plant cell membrane, so it's within the plant apoplastic space, is the hostorium. Now, the hostorium is the primary infection structure through which the pathogen secretes effector molecules in the host to suppress host immunity. And it's also the main feeding structure through which it absorbs nutrients and water from the host. So if you look at the typical life cycle of the pea powdery mildew, you will see that it's composed of 4 well-defined infection stages. So in the first stage, when a conidium lands on the surface of the leaf, it germinates, produces a germ tube. And at the tip of the germ tube, it produces this flattened lobe-like structure, which is known as the aprosorium. And through this aprosorium, it is able to penetrate the host cell wall. And also it helps to attach to the plant leaf surface. So this stage, which is known as the penetration stage, typically occurs between 9 to 12 hours post-infection. Now, if the penetration is successful, then this penetration peg invaginates the host cell plasma membrane and develops into this infection structure, which I just mentioned, the hostorium. And the primary hostorium fully develops for about 24 hours post-inoculation. And it's the main site through which it secretes effectors into the host, and also the feeding structure through which it absorbs nutrients. The third stage involves colony expansion, where you have extensive mycelial growth on the leaf surface, as well as production of secondary hostoria. And by about 5 days post-inoculation, you'll see that the fungus reproduces asexually by producing these stalk-like structures, which are known as the conidiophores, which bear conidia at their tips. And these conidia are readily dispersed by wind to initiate new infection cycles. And by about 7 to 10 days post-infection, you can visualize the powdery mildew disease symptoms on the leaf surface. Now, the disease can be controlled through the application of fungicides, but this method of control is not very environmentally safe or sustainable. So an alternate and more sustainable approach is to breed for genetic resistance. However, so far, very few resistance genes have been identified in agronomically important legumes. For example, if you consider pea, so far only 3 resistance genes have been identified, ER1, ER2, and ER3, of which ER1 has been extensively used in breeding programs because it provides very early penetration resistance. However, a breakdown in ER-mediated resistance has been observed against different isolates of the pathogen. And in many cases, the expression of resistance is temperature dependent. So there is a need to identify novel sources of resistance against this disease, which is where our research program comes in. The goal of our program is to identify novel molecular targets to engineer durable resistance against legume powdery mildews. And for this, a better understanding of the molecular mechanisms underlying this host pathogen interaction is essential. So for this, we have 2 major objectives in the lab. One is to identify the host resistance and susceptibility factors, and the other one is to identify pathogen virulence determinants. So in terms of host resistance, we are focused on investigating the role of secondary metabolites and sugar-related mechanisms with respect to powdery mildew resistance. And in terms of identifying susceptibility factors, we are focused on identifying those factors that can suppress the host immune response, as well as those that can impact fungal sporulation, with the idea that if we limit sporulation, we can limit the spread of the disease. The other objective is to identify the pathogen virulence determinants, particularly the effector molecules and their targets in the host, because they can dictate the outcome of this interaction. So in our lab, we use both P and Medicago as model systems to address our research questions. So today I will discuss some recent results that we have obtained on the project where we investigate the role of secondary metabolites in powdery mildew resistance. And credit for this work entirely goes to Arunima Gupta, who is a senior PhD student in the lab. So Arunima decided to use Medicago truncatula as the model plant for her study because of its inherent genetic variability or diversity in its germplasm, especially when it comes to powdery mildew resistance. So you have resistant, moderately susceptible, and susceptible genotypes that can be used to dissect the molecular basis of powdery mildew resistance. So you can see phenotypically at 12 days post-infection with Erysipeus piscis, the highly resistant genotype A17 shows no disease symptoms. The moderately susceptible R108 genotype shows mild to moderate symptoms, whereas the susceptible DZA genotype shows severe symptoms. And at the microscopic level, you can see that the pathogen growth is arrested at the aprosorial stage, which is the pre-penetration stage in A17. But the fungus is able to complete its life cycle on both the R108 as well as DZA. But the development of the pathogen is delayed in the case of the moderately susceptible genotype, and also fewer reproductive structures are found on R108 as compared to DZA. So a previous study had identified REP1 or resistance to Erysipeus piscis race 1 as the resistance gene against a Medicago truncatula isolate of E. piscis. So they're shown that this gene is a CCNB-LRR type of R gene, and that it's constitutively expressed in the highly resistant genotype A70, but not in a susceptible Medicago genotype. And also it's not induced in response to infection. So But the mechanism that or how this gene confers resistance to powdery mildew was not uncovered. So we wanted to explore, how REP1 can mediate resistance. So for that, first we looked at the expression of REP1 in the moderately susceptible and susceptible genotypes, and what we found was similar to what they had found. A17 is constitutively expressed in the resistant genotype, moderately expressed in R108, and little or no expression was observed in the highly susceptible genotype. But to confirm whether REP1 can also confer resistance to the P isolate of EPC, we transiently knocked down REP1 levels in the highly resistant A17 genotype and then looked at the pathogen phenotype. So what we found was that as opposed to the GFP silenced control lines, where the growth of the pathogen was mainly arrested at the aprosorial stage, on the REP1 silence lines, we were able to see that the pathogen can now penetrate, form a functional hostorium, and also develop hyphal structures by 48 hours post-infection. So this indicates that REP1 can confer resistance even against the P isolate of EPC. Now, a previous RNA-seq analysis study in our lab had suggested the potential involvement of the isoflavonoid biosynthesis pathway in powdery mildew resistance. So what are isoflavonoids? They're a subclass of the flavonoid group of secondary metabolites that are synthesized from the phenylpropanoid pathway. And they are mainly produced by leguminous plant species, and some of them are known to have antimicrobial properties. So we wanted to explore the role of this pathway in powdery mildew resistance. So when we looked at the expression of the individual genes in this pathway, we found two things. One is that a greater number of these pathway genes were upregulated in the resistant genotype as compared to the susceptible genotype in response to powdery mildew infection at 1 day post-inoculation. And secondly, in cases where it was induced in both genotypes, the magnitude of expression was always higher in the resistant genotype. And this you can see which I've shown here, represented here based on the color. So red indicates higher expression and blue indicates lower expression. So for her study, Arunima decided to use the highly resistant genotype A17 as well as the moderately susceptible genotype R108. And what she did is she followed the change in the alterations in gene expression of the pathway genes, as well as the accumulation of the isoflavonoid metabolites until 24 hours post-inoculation. And this time point was selected because at this time point, as we can see in the resistant genotype, the fungus is arrested at the aprosorial stage. But in this moderately susceptible genotype, it's able to penetrate. You can't see it very clearly here, but it's able to penetrate and form a functional hostorial complex. And this is indicated by the initiation of a hyphal peg on the surface. So this indicates that there's a functional hostorium. So what she wanted to see is if there was any difference in the kinetics of the expression of these genes and the accumulation of the metabolites between the resistant and the moderately susceptible genotype. So when she looked at the temporal expression of these pathway genes up to 24 hours post-inoculation, she found that majority of the genes that were encoding enzymes involved in the conversion of these isoflavones to the isoflavonoids were induced early in the resistant genotype, mainly between 6 to 12 hours post-inoculation, which are the time points during which the pathogen attempts to penetrate the host cell wall. Whereas in this infection-dependent expression of some of the same genes were slightly delayed in R108, occurring predominantly between 12 and 24 hours post-inoculation. The other thing was the genes encoding enzymes involved in this final steps, that is conversion of the isoflavone to the end product of the pathway, which is medicarpin, were much higher expressed in the resistant genotype and to a lesser extent in the moderately susceptible genotype, particularly if you notice IFR, which is isoflavone reductase. It's a key gene involved in this final step, was highly upregulated in A17, but not as much in R108. So based on this, we could conclude that isoflavonoid pathway genes are induced earlier in the resistant genotype in response to EPC infection. Now, to see whether this translates into metabolite accumulation, she performed a temporal expression analysis and quantified the accumulation or the levels of these different isoflavonoid intermediates, as well as the end product medicarpin in the two genotypes in response to infection. So in general, what she found was all of these metabolites were induced upon powdery mildew infection in both genotypes, but the magnitude of induction of the different metabolites varied between the two genotypes. So here in gray, I'm showing you those intermediates and precursors that are directly involved in the synthesis of medicarpin, which is the end product of this pathway. So what she found was in the isoflavone intermediates such as diadzine, formononetin, and one of the immediate precursors to HF were significantly, the levels of these compounds are significantly higher in the susceptible genotype, whereas the immediate precursor to MF and medicarpin, as well as the flavonone precursor liquidin, accumulated earlier and to higher levels in the resistant genotype. So what this indicated to us was that the conversion of these intermediate compounds to medicarpin occurs more efficiently and more rapidly in the resistant genotype. So the temporal expression analysis, as well as the metabolite accumulation, suggests that medicarpin, which is the end product of this pathway, is induced earlier in the resistant genotype upon powdery mildew infection. Now, since IFR, which was the key gene involved in this final conversion to medicarpin, showed differential expression between the two genotypes, we wanted to check if we transiently knock down the expression of IFR in the resistant genotype, whether we would see a breakdown in this powdery mildew resistance phenotype. So indeed, when we transiently knock down IFR gene in A17, we found an increase in the pathogen load, which was estimated through quantification of the RSF-PC18S ribosomal RNA transcript accumulation. And when we looked at the pathogen growth under the microscope, we could now see that as opposed to the GFP silence control plants, the pathogen on the IFR silence lines were now able to penetrate, form a functional hostorium and a hyphal peg. So based on this, we could conclude that transient silencing of IFR in A17 leads to a breakdown of powdery mildew penetration resistance. Now we next wanted to see if we overexpress this pathway. Can we mute people who are not presenting? Okay. So next we wanted to see if we overexpress this pathway, whether we would be able to enhance the resistance of the susceptible genotype. Now, typically MIP transcription factors have been shown to activate multiple genes in the secondary metabolite pathways in order to coordinate their expression and also initiate the synthesis of these secondary metabolites. So in our previous RNA-seq study, we had identified a MIP transcription factor that showed higher expression in the resistant genotype, but not in the susceptible genotype upon powdery mildew infection. And interestingly, this MIP shows 87% sequence identity to a soybean MIP, which was previously characterized as a positive regulator of isoflavone synthesis. So we decided to overexpress this MIP transcription factor in the moderately susceptible genotype and see if that can impact or enhance the powdery mildew resistance of the susceptible genotype. So indeed, when we overexpress, we found an increase in the transcript abundance of MIP, and we also found that this MIP localizes to the nucleus of the R108 leaf cells. And we also found that overexpression of this MIP could increase the expression of many of the isoflavonoid biosynthetic pathway genes. Whether this translates into metabolite accumulation, to check this, Arunima quantified the levels of the different isoflavonoid intermediates, and what we found was that in the MIB overexpression leaves, medicarpin and formononetin levels were significantly higher. So to see if this then impacts powdery mildew resistance, we quantified the percent of conidia that were able to produce hyphae, and what we found was that a greater percent of conidia were arrested at the aprosorial stage in the MIB overexpression lines as opposed to the empty vector controls, indicating that MIB overexpression can not only enhance isoflavonoid accumulation, but also confers powdery mildew resistance. Now, to further prove the role of this pathway in powdery mildew resistance, we decided to exogenously add or treat these R108 leaves with the different isoflavonoids and see if they could confer resistance to powdery mildew. So, and how did we quantify? We quantified the fungal load by quantifying the expression of the RSF-PC18S ribosomal RNA. And what we found was that if we pre-treat the R108 leaves with diadzine, formononetin, 2-MF, and medicarpin, there was a significant decrease in the fungal load at 48 hours post-inoculation. So this suggests that pretreatment with medicarpin or the metabolites that lead to its synthesis. So if you notice diazine, formononetin, and 2-MF, they are involved in the direct synthesis of medicarpin. So pretreatment either with the end product or the metabolites that lead to its synthesis can confer resistance to powdery mildew in R108. So taking all this information together, we concluded or hypothesized that the isoflavonoid pathway, and in particular medicarpin, may be involved in powdery mildew resistance in Medicago truncatula. So to further probe the antifungal role of medicarpin in powdery mildew resistance, we treated the R108 leaves with different concentrations of medicarpin to see how it can impact the pathogen load. So what we found was that medicarpin pretreatment can reduce powdery mildew growth in a dose-dependent manner. And it's important to note that at this concentration, that is the highest concentration, there were no visible signs of cytotoxicity observed in the Medicago truncatula leaf cells, as evidenced either by trypan blue staining, which stains for dead cells, or through DAB staining, which stains for hydrogen peroxide. And when we looked under the microscope, we could and quantify the fungal growth, we found that a greater % of conidia were arrested at the aprosorial stage when we treated the leaves with medicarpin as compared to the mock treated leaves, where we could see extensive hyphal growth. So we also wanted to check next whether there are differences in the spatial dynamics of this particular medicarpin in response to powdery mildew infection. So for this, we synthesized a fluorescent tagged version of medicarpin, and this work was done in collaboration with Dr. Atul Goel at CDRI Lucknow. And we then infiltrated this fluorescent tagged version of medicarpin into R108 and visualized the fluorescence under a confocal microscope. So at 3 hours post fluorescein tagged medicarpin infiltration or mock treated infiltration, where we infiltrated just fluorescein alone. So this was prior to inoculation. We found the fluorescence was uniformly distributed across the epidermal cells. However, at 24 hours post-inoculation, we can observe that this fluorescence was concentrated in a sort of a ring-like shape right directly beneath the fungal aprosorium. And in cases where the hostorial complex was formed, it was also surrounding the hostorial complex. But in the mock treated leaves, majority of the infection sites did not show such a pattern. So this was quantified, and this further confirmed that 80% of the infection sites showed this infection localized accumulation of the fluorescently tagged medicarpin, whereas only about 20% of the infection sites showed this kind of accumulation in the mock treated leaves. So what this indicates is that exogenously infiltrated medicarpin localizes at the powdery mildew penetration site, and this localized accumulation may be important for this early powdery mildew resistance. So next, we wanted to see how does medicarpin confer this resistance against powdery mildew. So for this, we detected the hydrogen peroxide accumulation in response to powdery mildew infection after medicarpin treatment, because we know that hydrogen peroxide accumulation is one of the earliest responses of the plant to pathogen attack, which ultimately leads to a HR-like response that can lead to cell death. So firstly, we found that fewer infection sites showed DAB staining, which is used to indicate the hydrogen peroxide accumulation in the medicarpin treated leaves compared to the mock treated leaves. However, at infection sites where the DAB staining was visualized, we found two different patterns. One pattern was where this DAB staining was visualized directly under the fungal aprosorium, and this mirrored what we had seen with the fluorescent medicarpin. And in the other case, it was present in the entire infected cell. So while the percent of conidia that showed this ring-like structure was similar between medicarpin and mock-treated leaves, we found that 43% of the conidia showed this entire cell DAB staining, as opposed to in the medicarpin-treated leaves, as opposed to only 2% in the mock-treated leaves. So this indicates that medicarpin treatment can enhance the infection localized hydrogen peroxide accumulation, and this was corroborated by performing the hydrogen peroxide quantification assays. So this indicates that this localized accumulation of hydrogen peroxide may lead to a post-hostorial hypersensitive response that leads to death of that infected cell, and that's how it can restrict the growth of the pathogen. But in addition, we also found that in the medicarpon-treated leaves, fewer conidia developed this primary hostorium as compared to the mock-treated leaves, indicating that medicarpon treatment, in addition to providing this post-hostorial resistance, can also provide penetration resistance against powdery mildew. Now, the phytohormones, salicylic acid and jasmonic acid, are known to play very important roles in pathogen resistance. So we wanted to see if powdery mildew infection can alter the levels of these phytohormones over the course of infection, and whether there are any differences in the accumulation between A17 and R108. So what we found was that SA levels were induced early and to higher extent in the resistant genotype as compared to the susceptible genotype, where it was actually suppressed at the later time points. Whereas JA levels were also increased in A17, but slightly delayed, and not so much in the susceptible genotype. So we wanted to see if medicarpon pretreatment can alter this infection-dependent accumulation of these phytohormones. So we designed an experiment where we infiltrated medicarpon into R108 leaves, and then after 3 hours of infiltration, we inoculated the plants with powdery mildew, and then observed the leaves at 48 hours post-inoculation. So what we found was that at 3 hours post-inoculation, we found both medicarpon levels to be significantly higher, as well as the SA levels. This indicates that medicarpon treatment can induce synthesis of SA, but when we looked at the JA levels, there was no difference. So it does not induce JA levels. But at 48 hours post-inoculation, medicarpon levels are significantly higher, but when we look at SA levels, you'll see that there is an increase in the medicarpon-treated leaves, but the difference between medicarpon and mock-treated leaves was not significantly different, possibly because powdery mildew infection can also induce SA levels in the mock-treated leaves. So maybe that's why the difference was not very significant. But overall, we can conclude that medicarpon treatment can induce synthesis of SA, but not JA. So we wanted to see if this increase in SA synthesis is due to an increase in the expression of the SA biosynthetic genes. So we followed their expression at 1 hour post-medicarpon treatment, 3 hour post-medicarpon treatment, and then after that at 48 hours post-inoculation with powdery mildew. So when you look at PAL1 and ICS1, which are the SA biosynthetic genes, PAL1 was induced at 1 hour, but not at 3 hours after medicarpon treatment. But ICS1 was not altered at both time points. So this indicates that the SA synthesis that is boosted by medicarpon treatment may be dependent on the PAL pathway, and that the gene expression may precede the metabolite accumulation. However, if you look at 48 hours post-inoculation, both PAL1 as well as ICS1 was significantly induced, indicating that both pathways may be involved in SA synthesis in response to powdery mildew infection. We also looked at the expression of the SA signaling markers to see if SA signaling is active. And although their expression of these PR genes or pathogenesis-related genes, which are well-known SA signaling markers, did not occur at 1 hour post-medicarpon treatment, many of them were induced either at 3 hours and or 48 hours post-powdery mildew inoculation, indicating or correlating with the resistance phenotype that we had seen. So next, we wanted to see if medicarpon treatment can also alter the accumulation of its own isoflavonoid precursor metabolites. So for that, we quantify the levels of the different isoflavonoid metabolites that lead to medicarpon synthesis. So in general, we find that the levels of these metabolites did increase upon medicarpon treatment, but significant difference was only observed for formononetin and for liquirtin. So there is a based on this, we hypothesize that potentially medicarpon can induce, positively induce the its own synthesis. So when we looked at the expression of the key isoflavonoid biosynthetic genes upon medicarpon treatment, we found that IFS1 levels, which is the gene that encodes the enzyme that is involved in the first committed step towards isoflavone synthesis, was induced at 1 hour, not at 3 hours, but again at 48 hours post-inoculation. And this expression pattern seemed to mirror what we had seen for PAL expression, indicating that medicarpon treatment may potentially positively regulate its own synthesis. Now, since we had seen that medicarpon could induce SA levels, in literature it's known that SA treatment can induce the expression and the accumulation of flavonoid metabolites. So we wanted to see if SA treatment could then induce this medicarpon synthesis. So we looked at the promoter of some of the key genes involved in this isoflavonoid biosynthesis pathway, such as IFS and IFR, and we found some SA-responsive cis elements to be present in them. So we then treated R108 plants with SA and then checked whether it could induce the expression of some of these genes, and we found that indeed it was able to induce the expression of IFS as well as IFR. So next we checked whether it could induce accumulation of medicarpine. We found that at 3 hours post SA treatment and at 48 hours post powdery mildew inoculation, levels of SA as expected were higher in the SA treated leaves compared to the mock treated leaves. But when it comes to medicarpine, there was a slight increase in medicarpine levels at 3 hours post SA treatment, but this difference was not significant. Whereas at 48 hours post inoculation, this increase in medicarpine levels was significant. This indicates that SA treatment can also induce medicarpine synthesis, but only in the context of powdery mildew infection. We also looked at JA levels. We found that at 3 hour SA treatment, JA levels were significantly downregulated, possibly reflecting the antagonism between the two phytohormones. But at 48 hours post inoculation, there was no difference between SA treated and mock treated leaves, suggesting that JA levels are induced in response to powdery mildew infection. So based on this, we could conclude that SA treatment can also induce medicarpine accumulation upon powdery mildew infection. We also wanted to check whether SA treatment can alter the levels of the isoflavonoid intermediates that lead to medicarpine synthesis. And we found that SA treatment could only significantly induce levels of 2-MF, which is the immediate precursor of medicarp, indicating that SA treatment may actually lead to an increase in medicarpine levels through increasing levels of its precursor 2-MF. Lastly, we wanted to see whether the activation of these two, the isoflavonoid and the SA pathways, was dependent on REP1, which is the R gene that was previously identified in Medicago truncatula. So we looked at the expression of the isoflavonoid biosynthesis genes, as well as some SA biosynthetic genes, as well as regulators of SA biosynthesis in the REP1 silenced lines compared to the mock silenced lines. And what we can see is that the isoflavonoid biosynthesis genes were significantly repressed in REP1 silence lines as compared to the controls. However, this same result was not seen for the SA pathway, except for EDS1B, indicating that even in the absence of infection, REP1 might regulate the levels of the isoflavonoid pathway, but it's possible that only in the presence of infection, it can regulate the SA pathway. So based on our results, we have now proposed a model to explain the early powdery mildew resistance that is observed in the highly resistant genotype of Medicago truncatula, in which the R gene REP1, upon recognition of the powdery mildew effectors or effector function, can quickly activate isoflavonoid biosynthesis, possibly through the MIP transcription factor. And this leads to a localized accumulation of medicarpine, but it can also independently also activate the SA and JA pathways, and together medicarpine as well as SA can lead to an induction in hydrogen peroxide accumulation at the infection site, which could then lead to a post-hostorial HR-like response, or the localized H2O2 accumulation could also lead to penetration resistance. Now, although we have shown that exogenous medicarpine treatment can boost SA levels and exogenous SA treatment can boost medicarpine levels, it would be interesting to test whether this phenomenon also occurs during the natural course of infection. That is, whether endogenous levels of medicarpine can boost SA or vice versa. So we have some experiments that we have planned for our future directions, where we're going to quantify the levels of isoflavonoids in SA deficient mutant plants, and then quantify levels of SA in isoflavonoid deficient mutant plants. Second is to quantify isoflavonoids and SA in susceptible plants that are overexpressing the REP1 gene to see if there's a connect between the R gene and these pathways. And thirdly, many of these isoflavonoid biosynthetic enzymes are known to be localized on the outer surface of the ER, and they form these clusters, what are known as metabolons. So in preliminary experiments, we had seen that the R gene REP1 localizes to the ER, and in response to infection, to the extrahostorial membrane, which is also known to be ER derived. So it'll be interesting to see if Rep1 and these isoflavonoid enzymes co-localize, and that is how Rep1 can activate this pathway. So these are some of our future directions. So with that, I'd like to just acknowledge the people who have contributed to this project, particularly Arunima Gupta, who spearheaded this project and has performed a majority of the experiments. And I'd like to thank our collaborator, Dr. Atul Goel, for synthesizing Medicarpin and the fluorescein tagged Medicarpin, and Dr. Nirpender Singh and Neha at ATPC for the metabolite quantification. And I also like to thank our funding sources, SERB, DBT, and co-funding that we get from RCP. So with that, I'll end my presentation. Thank you all for your attention.
Very nice. So I think if there will be any question.
So Divya.
Okay,
yes.
Madam will be asking.
Yeah. Very nice talk. So maybe I don't know whether I have missed it. You have told it, but I was just wondering that you already have done previously calculation of these effectors also for this particular pathosystem, right?
Yes.
So I was wondering, do you know which is the effector actually is targeting this Rep1?
No, so that's an interesting question, but we still don't know. We're in the process of identifying which effectors could target or be targeted by Rep1.
Okay,
we don't know.
Nice talk, Divya.
Thank you. Yeah.
We'll be meeting again during the panel discussion. Now the talk will by Dr. Senthil from NIPGR. Senthil sir.
Yes, I'm here.
Namaskar, namaskar. You please say.
I'm going to share my screen. Morning, all of you. Suvendra, can you please confirm whether the screen is visible and I'm audible?
Yes, it is perfect.
Morning, everyone. Again, I'm privileged to be part of this team and especially part of this core team for quite a bit of time. Thank you so much for organizing this, Subhadeep mainly. So today I'm going to talk about sweet revenge, that is the role of sweet class of sugar transporters. Our lab has been working in this transporters for the past 9 years. And we have made several headways. Today, I think some of you might know part of these results, but today I'm going to mainly talk about the comments that we got from the reviewers, how we addressed it, and how the story is now reshaping. So that's the part I'm going to explain it. So the first thing I'm going to explain is a brief of the introduction. We all know that sugars are an important part of carbon source for pathogens. And that's the reason why they come to the plants. And this niches includes philosphere, apoplasts of both the leaf and other organelles, phloem, xylem, and others, of which the leaf apoplast is one of the major source for a lot of aerial pathogens, mainly the bacterial pathogens that I'm going to talk today. I'm also going to introduce a bit about because the whole study uses the host and non-host pathogens to make the classification clear. I would like to introduce the non-host resistance concept. So this most of us knows, but just briefly, a pathogen X that lands on a plant 1, which can cause disease, is called host pathogen. But the same pathogen, if it lands on the plant 2, and that plant is able to effectively provoke a defense response. In this case, I'm just highlighting the hypersensitive response. So which is basically a non-host pathogen for that plant. So the vice versa also could be possible. So whenever I say about the host pathogen, I'm talking about a pathogen that can potentially cause disease. When I'm talking about the non-host pathogen, I'm talking about the one that the plant can effectively mount. And in the past, MPMA has found that this non-host resistance topic is one of the top 10 unanswered questions, and that needs to be followed up. So it's becoming an important emerging area, and MPMA as a whole is focusing on this. Especially they welcome the articles and also the area interest, research interest in this area. So in this study, the non-host pathogen that we will be using as of 2 kinds. I'm focusing on both the pathogens, although I will not be distinguishing this often in the slides. Basically, to just give a glimpse, type 1 non-host resistance does not produce a visible symptom. And however, the other pathways that are involved in the defense takes care of this pathogen. Type 2 non-host pathogen typically produces the hypersensitive response. So in this case, the tobacco pathogen, the tobacco wildfire pathogen produces the hypersensitive response in Arabidopsis, which is non-host plant. And in this case, the Pseudomonas phaseolicola, which is a bean pathogen, that produces that cannot multiply in this plant, is a type 1 pathogen. And of course, we know that the host pathogen typically produces the disease-induced cell death. And in Arabidopsis, it is typically yellowing followed by chlorosis and mottling, and also subsequently the cell death whenever the bacteria enters into the necrotrophic phase. And just a brief introduction about this apoplast. We know that apoplast is a major battleground for foliar bacterial pathogens. And just to emphasize that, so I will just overly explain it. So the labeled bacteria that here clearly shows that they are confined to the apoplast. So it's to be noted that these pathogens never enter into, these bacterial pathogens never enter into the cell, except that they produces the type 3 secretion system to insert, either to suppress the plant immunity or to bring the nutrients back into the apoplast on which they grow. So these pathogens are confined to it, which means that they are desperate, and their only way is to get the nutrients into the apoplast for them to feed on. Otherwise, there is no way. So initially, there will be some initial nutrients that are as a part of apoplastic exchange can be useful. But later on, once the population grows, there must be a need for this pathogen to get the nutrient out. That is where this whole project and the involvement of sweets comes in. We know that this host pathogen growth in the apoplast can be widely there, and the non-host pathogen growth in this is almost nearly minimal in the inoculated leaves, as shown in these two images. The reason I'm trying to explain this is that if you take an apoplast from a plant that is not infected, that apoplast has a minimal level of nutrients, basically the level of nutrients. And however, if you take the apoplast from a host pathogen inoculated plant, it contains a lot of nutrition, including minerals and many other nutrients. However, in the apoplast of the non-host pathogens has a very limited amount of nutrient, which is much less than even a control apoplast, which is almost equal to that of a minimal medium otherwise that it grows, which means that the non-host pathogen, when infected, the plant is able to successfully withhold the nutrient as well as dispense of the nutrients available at the apoplast. So this was very evidently proven by several Saxon papers earlier. So basically, this two pictures shows that the control plant where there is a clear non-host resistance exists. On the right-hand side, the NBSQS, which is a gene that is involved in membrane sterol composition, whose alteration or disruption leads to leak of multiple number of nutrients into the apoplast. So you can clearly see that the host pathogen of this Pseudomonas aeruginosa goes very nicely. However, all the three types of non-host pathogens containing pathogens, basically the non-host bacteria, is not able to grow in the left-hand side where there is where this gene is intact. So you won't see a growth of the bacteria. However, the moment that gene that leaks all the nutrients out into the apoplast is there, then you will see all the three non-host pathogens also growing. Almost some of them is as equal as that of the host pathogen. So which basically again emphasizes the point that regulating nutrient release into the apoplast can be a good control strategy for the plant. And hence we aptly formed a hypothesis that can the plants limit the sugar availability at the apoplast and thereby form a parallel immune pathway that can limit the bacterial growth as well as thereby preventing the infection. So largely the sweet class of transporters have been explored as a target of host pathogen. So this has been much explored until recently, even to the rise the pathogens, bacterial pathogens. However, our concept is much different from that. Basically, we are trying to see whether these sweet transporters can become a player as a part of plant immunity rather than becoming a susceptible factor for the pathogens. And of course, it was part of the plant immunity. That's why even the pathogens also started targeting these pathogens. Fortunately, so far, there is no report that shows that these pathogens are able to target sucrose transporters. They have been well known for targeting the glucose transporters. Now, so in this talk, I'm going to take one class of, that is the sweet class of transporters, and whose role in playing defense against the bacterial pathogens. This work was entirely done by Uruj Fatima, and the system we are using is the Pseudomonas syringae and Arabidopsis as a model system. So these sweet class of transporters are divided into majorly 4 clades, and mainly these have 2 broad category of substrates, that hexoses and sucrose. And these there are 17 sweet transporters in Arabidopsis, and these also plays multiple roles, not just in plant microbe interactions. They have a multiple physiological roles in the plant. This picture shows its location either in the membrane, plasma membrane, or in the vacuole, the tonoplast membrane. So these are all the different locations of different transporters and their functions. They are involved in pollen development, seed development, and also there are also other roles in freezing tolerance, like other abiotic stress tolerance as well. They play a role. So these are all part of a regular metabolism maintenance transporters, these sweet class of transporters. So when we took all the 17 transporters and then did both the transcript profiling as well as the reverse genetic screening of all the mutants, we found that there is uniqueness to, there are 3 transporters, that is 80 sweet 11, 12, and 15. We found that the mutants of 80 sweet 11 has less bacteria, and the mutants of 11 and 12 had higher levels of bacteria, which means that whose knockout has led to probably more nutrients and thereby presence of more bacteria in the apoplast. We have also proven the same thing by using the double mutants of 11-12, 11-15, and 12-15, and triple mutants like 11, 12, and 15. So the interesting thing is, whenever you have whenever you work with a mutant background that has 11 in mutation, so they are all almost either equal to that of wild type, even if it is a triple mutant. As long as 11 is mutated, there is lesser level of bacteria. As long as there is 12 or 15 to some extent, but 12 constantly, the resistance is compromised, and you see more bacterial growth for this PSP, which is basically the bean pathogen, which is a non-host to Arabidopsis. And it could even go to the extent of causing symptoms, which is very rare for a non-host pathogens, by compromising 2 genes. Having a symptom developed is a higher level of vulnerability to the plant, almost like a host pathogen. Now, we also complemented these mutants to prove the same point. So similarly, whenever we complement either with 11 or 12, the reverse phenotype was able to obtain. So whenever we complement with the 11, this complemented line was showing the susceptible, complemented with the 11 in the double mutant, it was showing the susceptible response. It was almost equal to that of 12. Whenever 12 was complemented, there was a slight resistance because there was 11 that was in play. So that is the reason that the complementation results also showed the results that what we have proven in the transcriptomic as well as the mutant analysis. Now, going forward, we were trying to understand what is happening to this. So these are all gas fusion proteins indicating the accumulation of 11 or 12 proteins in the. So you know that these are all localized to plasma membrane and to tissue. So under the normal conditions, 11 proteins are but not the
Some background noise is coming, Senthil. Can you somebody mute? Excuse me, somebody should have to mute.
Please mute.
Okay, thanks. So if you look at this picture under the mark, so the fusion protein, whenever it is driven by a 11 promoter, you are able to see a normal expression and accumulation of this sweet 11. However, under the normal conditions, the sweet 12 is not expressed. Whenever you inoculate this plant with the host pathogen, you can clearly see at 0 dpa there is a expression, but the expression levels of 11 goes down. However, the levels of 12 increases, both at 36 and 48 dpi. Although it slightly goes down at 48 dpi. The non-host pathogen, you clearly see, again, the suppression of this accumulation of 11 is much less, and it was increasing at 36 hours for 12, but again goes down after some time. So basically, it renders a point that the 11 protein is accumulated in the normal conditions, but not 12. After pathogen inoculation, the accumulation of 11 protein is reduced. However, the protein 12 is increased. This was also selectively proven by using the YFP assays that whenever we expose this to pathogens, both for this non-host pathogen as well as for this host pathogen, we could able to see the targeting of these 12 protein into the membrane. And it was very clear from the experimentation that there is a differential regulation in response to the pathogen between both 11 and 12. Now, so we went ahead and quantified the sugar content, and it was showing very clearly that there is the level of sweet, the mutant, the sugar levels in the 11 mutation as well as the 11 plus double mutants was less, whereas the sugar levels, especially the sucrose levels, was much higher in the single mutant. So basically, to summarize this point, that whenever there is a mutation of 11, the apoplastic sucrose levels were less, and the apoplastic sucrose after the infection was not changed. The pathogen multiplication was less. Eventually, when there was less sugar, there was less pathogen multiplication. In the 12 mutant, again, there was no change, and in the control conditions, whenever you infect the plant, it became more, and thereby it produced, it allowed more bacteria to multiply. In the 11 and 12 mutant, it was almost similar to that of 11 mutants. So the apoplastic sugar at the control was less, and whenever there was infected, again, there was less, and the pathogen multiplication was much less, even much lesser than that of the wild type. Now, so these points were not fully convincing the reviewers, so they were asking us to do a lot of other experiments, of which I'm explaining two experiments that basically they wanted to have in vivo or a real-time estimation of such fluxes. So it should not be a stagnant pool or the sucrose pool that is probably contributing and beyond which these transporters are not actually playing the role. So that was the major comment. So to address this comment, we had two important experiments designed. The upper one, that the graph involves quantification of luminance assay. Basically, there is a bacteria, Rhizobium leguminosarum. So this bacteria has a sensor which can sense and illuminate against the sucrose levels if it is in the presence of sucrose. So we inoculated that on a real-time basis and then estimated the depletion or alteration of sucrose in the apoplast. So the assay clearly showed and distinguished that whenever you have a higher, just the sucrose only is there, the illumination was much higher. And the mutants of the 11, both of them, either in the double mutant or in the single mutant, the sucrose levels then in terms of luminescence was much less. And we have quantified this for a much larger levels, larger time duration, and we found that dynamic regulation of this is happening in the wild type, and hence the non-host resistance was able to achieve in that. In the mutants, that it could not happen, and hence the observed phenotype was observed. The second experiment we did was a simple labeling of this bacteria with the GFP UV construct, where it can fluoresce under the when it exposed to UV. And this was also on a real-time basis estimated. And I think this movie is not going to play, but I'm just explaining that. So the 11 mutants, as you can show here, the non-host pathogen growth was almost nil. Even the host pathogen did not grow much, whereas the 12 mutants supported on a real-time basis much higher growth. So both the sugar depletion and the real-time monitoring of this bacterial growth and its multiplication clearly convincingly proven the previous results, and it was able to say that now the dynamic regulation of the sugars by these 11 and 12 transporters differentially has been the reason for either reduction or increase in the bacterial pathogen in the apoplast. Now, the alternate experiment that also we did again to support the same concept was to supply externally the sucrose into the apoplast in those mutants. To just briefly say that the resistant phenotype of this 11 and then the 11-12 mutant, wherever there is a mutation of 11, the addition of sucrose clearly compromises this resistance, clearly indicating that it is possible that we are able to prove the concept by saying that the sucrose is the major key determinant that was playing all the role, not the other questions that the reviewers have asked to prove that whether it is not due to any other factors, either that could be back signaling of the sucrose signaling and others. So we could able to establish that it is the sucrose that is able to play the role. Now, just to briefly explain the mechanisms which we have started working for the past 1 year. So in collaboration with Sam's lab at ICGB, we were able to start working on these proteins. So you can see that it is sweet 11 and 12, and mainly I would like to draw the attention of this enlarged view where the sucrose could be docked to see where it was actually fitting. So mainly the interactive view also shown that we could able to predict the sucrose view, and also experimentally partly we were able to validate in a non-plant system, whose data I'm not showing right now. So this basically proven that there is a possibility that this 11 and 12 could differentially regulate this by forming either homo or hetero oligomerization. Because just to mind you that this 11 and 12 transport is decided by its oligomerization status to form the cavity to allow the sucrose to pass through. So the hypothesis that we had was that in order for a functional protein to form, transporter to form, the 11 should form its oligomerization with its own homomer, that is 11 and 11 or 11 and 12. So then both can be an independent transporters. However, since because of its highest homology and also the residual ideologies, identity, it's possible that this 11 and 12 could form a heteroligomer. So to test that, what we have done is that we have taken the sucrose deficient mutant. I would like to just draw the attention for only these two lanes, where you can clearly see that the sucrose deficient strains were not able to grow in those lines where the constructs with 11 and 12 were put together transformed. And that is mainly because of this heteroligomerization where the transport activity was hindered. And further, to answer substantially, to add the substantial evidence to that answer, radiator sucrose uptake assay was again done. I just like to draw your attention to the Vmax value of this last graph, that is the first graph, that is 43.1 versus the other Vmax values. Clearly again indicating that there is a possibility that these two proteins form heteroligomers, and thereby that might hinder the sucrose transport activity. And hence in the wild type, there is a possibility of bringing out the resistance for the non-host pathogens. Now, the other question that was clearly on the cards was that how much it is part of a regular immune pathway. So some of the initial experiments through the yeast library screening, as well as through some of the old evidences that was produced by one of the German groups who shared the data with us, has clearly showed the possibility of involvement of FLG22. And we could able to successfully show that the FLG22 is able to bind and regulate the 11 and 12 transporters. And now we have more experiments to do to clearly say how this regulation distinguishes this role of 11 and 12 as well as its interaction. And our just recent initial experiments points towards the status of phosphorylation of this 12, which could probably determine how that sugar transport should happen in response to which pathogen. And it could even be able to distinguish between the host and non-host pathogen infected plants. I hope I'll be able to share this story in the next time. So basically my point is we could able to clearly show that the differential role of 11 and 12 and whose regulation of sucrose at the apoplast is able to determine the disease resistance or susceptibility in Arabidopsis. However, you could clearly see that the non-host pathogens were still be able to grow to some extent, and so the reviewers were of the opinion that is this pathway of sugar limitation is complete. So we know that from our previous VIX-based screening of about 5,000 clones, there was another candidate that popped up in that screen was SUP2, and another sucrose transporter, which is again localized to the phloem. It is basically involved in the process of phloem uploading. So just to brief you that sweet 11 and 12 downloads the sugar into the apoplastic region of those C-filament companion cell complex. And thereafter this SUC2 loads that into the phloem region. So that is how the long distance transport of the sugar happens. So the hypothesis was that whether there is a simultaneous role for both 11 and 12 and whose regulation, and the SUC2 who can further deplete the sucrose levels at the infection site, namely the apoplastic site, could be the player. Thereby, the starvation of the pathogen happens, and the role of other immune pathways, namely the RAS or the antimicrobial compounds at the minimal level, was able to control the population and thereby eliminate the bacterial pathogen presence in the apoplast. So we just briefly had, so this now became a project on its own. I'm just showing only the brief results of this. We could clearly prove that there is a differential regulation of this SUC2 against the host and non-host pathogen. DC3000 is the host pathogen and PTA is the non-host pathogen. And we could also clearly show the accumulation of this protein is differentially there between the host and non-host pathogen, and which was not there in the mark. Basically, there was more accumulation against the infection, especially with the non-host pathogen infection, as you can see from the in the phloem basin region, especially, that this SUC2 is much highly accumulating, just like the one that you could see with the 12 protein. Now, we have also proven that through the in situ hybridization, that these mutants that are the SUC2 mutants, which behave much like a resistant type, were not able to accumulate the SUC2 transcripts, and the antisense lines, you can clearly see that this clearly shows that the SUC2 could be a regular player to explain much more depletion of the sucrose levels in the apoplast. So we took all the possible single and double mutants and also overexpression lines, both with the native promoter and also the 35S promoter. In nutshell, I just like to say that whenever we include SUC2 as a player, and coupled with the role of 11 and 12, we are able to completely explain the point that there is a possibility of depleting the sucrose at the apoplast. So just to brief the whole story that we got in this slide. So the sieve element and then the companion cell complex is the major complex that actually loads sucrose into the phloem region, whose the transport of the sucrose comes from the mesophil cells and then passed on to the phloem parenchyma, and then downloaded by these two transporters, that is 11 and 12, and also to some extent 13 and 15, into the apoplast of this region, that companion cell sieve element complex region. The blue colored sucrose transporters, that is the SUC2, is able to load the sucrose into the phloem for the long distance transporter. So note that the Pseudomonas does not infect the phloem region. So it clearly explains that the companionship role of this 11 and 12 regulation along with this SUC2. SUC2 is able to deplete the sucrose levels in a much faster way, as well as 11 and 12 regulation is able to not to allow the sucrose to be transported at the site of pathogen infection. And further, we were able to clearly show that compared to the single transport of any of this either 11 alone or 12 alone function, the heteroligomerization that is formed between the 11 and 12 transporters could be the major reason that could limit the availability of the sucrose at the site of infection. So this could largely prove our point. So I think with this I will close, and would like to thank all the lab members and also funding from NIPGR and DBT. And I would like to take up the questions, if any. I hope I was within the time. Thank you so much.
Very nice. So can we, I think now, sir, can we have the sir panel discussion at the time, or the question can be addressed to all along with Dr. Senthil?
Yeah, sure. Yeah, I think better after panel discussion, during panel discussion, because we are running out of time.
So now I think we have sir, Dr. Gopal, Dr. Divya, and Dr. Senthil. We can ask, discuss questions to them. Sir, I'll ask question to Dr. Senthil.
Yes, yes.
Dr. Senthil, how does this the non-host pathogen are recognized by the plant?
So we could still figure out based on that library, what is the polysome library, what we got from German group, that the FLG is one of the typical pathway, that FLS FLG recognition that is still on. That is basically the PAMP is still integrated and on. And so far we know that this is one pathway that is normally explained as a explained for a host resistance, is also operating under non-host resistance. Apart from that, there is a glycerol kinase which mediates a pathway parallel to this, has been shown to operate not normally with the host pathogens, but with the non-host pathogens. These are the two known ones that I could explain as a thing that how the pathogen is recognized and then the signal is passed on and how the immunity is brought about against the non-host pathogens. But it is a much unexplored area.
So I will ask one question to Dr. Divya.
Can I ask one add-on?
Please.
So, what's the fun of interaction of FLS2 and this transporters, sweet transporters?
So actually, this we didn't expect. We were doing the assays thinking that it may not interact, but both during the pull down and also this assays QIPs clearly indicate they interact. So we still do not know what actually it has to do. I thought there must be an intermediate involved, why it should go and interact directly with this one.
Because
so
both are like transport cell membrane localized.
Yeah, which is also difficult to explain. But actually right now we are deviating the story a bit by introducing this phosphorylation step. 2 weeks ago, there was a Nature Plant paper exactly on these two transporters explaining very similar to that of us by introducing the phosphorylation, but that was for an abiotic stress. So they clearly show and also throw a light in the same question what you have asked by introducing the phosphorylation state of it. The only difference is in their case, the phosphorylation makes the transporter to form a heteroligomeric state. But in our case, probably it's the opposite, maybe the dephosphorylation. So we are proceeding to identify a kinase. I think Anjali is able to get one kinase into a role to play. So we probably may not stick to that FLG mediated thing, although the evidences are up there to show that it is binding and there is a regulation possible. So we are moving towards more of that MAP kinase pathway, which finally probably phosphorylates or dephosphorylates the 12, not the 11, but the 12.
Yeah, but that should be independent of FLS2.
Yeah.
Most likely.
Can I ask a quick question to Senthil regarding the same topic? Have you tried the big one and back one? They are the co-receptors of FLS2. I think that should maybe
that's what Anjali is doing right now.
So,
so hopefully she will be able to throw better light into it. But for this paper, the reviewers are kind of convinced with the FLS2 assays, so that's going parallelly. But yes, I think I'm not I'm not able to explain directly about how FLS2 binding to a functional transporter is explainable, and if that is the case, why there is a distinction between host and non-host. I mean, there are a lot of questions. Yeah.
Because it has been postulated that all these receptors and co-receptors form these nanodomains on plasma membrane. So they bring in different interacting partners under each kind of resistance and bring in channels or transporters or co-receptors which are specific to that pathway.
Yes,
so
the backbone complex what you are the complex what you are suggesting is exactly what the one that is possible to explain it in a better way. Yeah, and she is working on that.
Thank you.
Hi, Senthil. I'm Sarup. I have one question. So in your suite 11 and suite 12 mutant, when you compare, so one of the phenotype I have seen that when you are making the double mutant, it looks like the wild type. So how would you explain the genetical way it is regulating them?
So it depends on how, so it is equal to wild type in response to a pathogen, especially the non-host pathogen. But if you just compare a regular phenotype, maybe in terms of either the starch accumulation, so it's not equal to starch accumulation is much higherly reduced in those mutants, which there is a clear distinction. Although such an accumulation does not translate into a bigger phenotypic differences. They all look normal. They flower at nearly the same time, not exactly the same time, but nearly the same time within a week interval. So the phenotype is equal to wild type only in response to its non-host pathogen. In other ways, wild type and the double mutants are much distinct.
Until I have a couple of questions for you.
Yes, sir.
So the suite 12 you're saying is going to compromise suite 11 by forming the heterodimers. Is that what your heterodimer?
That's what I'm saying. And sweet 12 is the one that's shown to be recently phosphorylated by drought stress, during drought stress.
Okay.
So the hypothesis more is that normally 11 is a functional transporter. Although 12 could also form a transport, do a transport activity, but it is not normally a transporter. It's more of a regulator.
I see. Okay. And the second question is, FLS2 mutants, are they compromised in non-host resistance at all?
Yes. So actually, it is very difficult to work with FLS2 mutants because they are a very strong phenotype. I mean, I don't think we can explain anything by creating a double mutant. I mean, she has done virus-induced gene silencing to explain this one, but none of the phenotype could overcome the regular FLS2 mutant phenotype. So, but in the overexpression lines of FLS2, we could see subtle differences to explain this point.
Okay. Very interesting, sir.
Thank you.
Very interesting.
Sir, can we say this cytokinin is somewhat similar to quorum sensing?
Maybe Sumendra. Down the line, maybe we'll find that, because what Gopal is finding recently is that stationary phase cultures of XOO seem to be making more cytokinin than the early exponential cultures, or even mid-log, early log or mid-log, even late log. Late log and stationary is where maximum cytokinin is made. So yeah, maybe there is a quorum sensing aspect of it that we have to think of down the line. Yeah, sure.
And more broader sense, why cytokinin has been selected as a signaling molecule in XO?
In any bacterium or even in plants?
Yes, sir.
How was it selected? We don't know, Sumendra. It's a very good question. Maybe, there was some time in, it's derived from a component of a nucleic acid. Maybe there is something with regard to cell division, that has, because cytokinin controls cell division in plants. And we think it is actually controlling cell division in bacteria, because there are some other mutants. When we knock out the alternate pathway for cytokinin, we actually see what are called long snaky-like cells, where cell division is actually stopped. So maybe there is something earlier. It must have happened very, very early on in evolution of cells, that they needed a molecule that is involved in making nucleic acids to control some aspects of cell division. I don't know.
Ramesh.
Yeah, Subhadeep.
This elongation in cells, actually, we see many times as a result of particularly the oxidative stress and stationary phase stress also. But the stationary phase stress will be different, because the cells which has a problem in the adaptation to stationary phase more becomes spherical rather than rod. And rod cell, more elongated cell arise due to problem in the reactive oxygen species balance. So we can check once in these mutants, what is the ROS levels at different phase of the growth. That will be one very interesting. And whether we can see that if we get some indication that there is some ROS connection, then we can add some antioxidative agent like citric acid or something, which can suppress that phenotype and see. So definitely this elongation happens due to stress. And if that connection is also not there between the that ROS and then we can see whether this this cytokine mediated signaling is affecting the FTSZ mediated septal formation and this thing. So in this this transcriptome analysis, do you see some cell division proteins or something like FTSZ
In the, see, we have done, as I said, there are two pathways.
Yeah.
Pathway 1 is what ZOQ is actually, what we call pathway 1 is what ZOQ is actually involved in. There's a second one, which is actually, XOO has a gene that is homologous to the rice protein, Arabidopsis protein called lonely guy. Arabidopsis in rice, that catalyzes the last step in cytokinin production. So when that is mutated, we get the snaky phenotype. So we think that both pathways are doing two different things. One is controlling biofilm, pathway 1 is controlling biofilm to lanthanate transition. The second pathway we think is controlling cell division. But we have to work it out. Maybe the next MIPAM will have more data about the second pathway. Then we can discuss it in more detail. Yeah, I must say to Suvendra's question that we really don't know why isopentenyl adenine is a signaling molecule. That matter, we don't know why. I don't know at least why auxin is a signaling molecule. Why acetyl homoserine lactone? Why homoserine lactone is a signaling molecule in bacteria? So what determine how were signals? How were certain signaling molecules identified early on in evolution as being important signaling as opposed to other things? So I really don't know, Suvendra. I should say no to answer, but I was trying to elaborate and try and do some hand waving. But it's
just a related point, a very generic point. So even prokaryotes are known to produce cytokinins, right? So how do you explain that? Okay, so you are working in the perspective of plant cytokinins against the prokaryote who might potentially produce cytokinin as well.
Actually, what we were all the time we have been thinking that bacteria that associate with plants produce cytokinin to modulate the plant. Okay. But what we are finding is that they are producing cytokinin to modulate their own physiology. So there's a role for cytokinin in modulating bacterial physiology. That was not known. So we are saying that bacteria make their own cytokinin to modulate their own activities.
And evolution, maybe what you said also earlier, that in evolution, maybe plants have hijacked that from the bacteria to have their own signaling.
Yeah, sure, definitely.
Because of the plastid and mitochondrial association, plants might have learned from bacteria to use that as a signal.
We know that nematodes also secrete cytokinins, right, to modulate the plant for gall formation.
Sure, they do that. But now there is evidence that dictyostelium also makes cytokinin, and that cytokinin controls physiology in dictyostelium. So cytokinin may actually be a signaling molecule in many different kingdoms, not just the plant kingdom, but also in the bacterial kingdom and in the protists. So, yeah.
So, another interesting thing maybe, shall I, or maybe you go ahead, sir.
Nandi sir, please.
Nandi sir, you go ahead, please.
Okay, actually, my question is to Sendil. Say, did you really confirm that this transporter SWET11, SWET12 is specific to sucrose, or it could be other molecule?
So that, this is largely a sucrose transporter, but now we know that it could also potentially transport GA. So this is not proven, but I don't see an association of
GA, gibberellic acid.
Yes. So
they're very structurally different.
Yes, so we are not clear about this. Is the at least the reviewers' comment. So I'm not sure how much it was experimentally proven, but yes, it could widen up a opportunity of having other substrates involved in this.
Yeah, because actually, if you see the non-host resistance, this glycerol metabolism also plays a very important role. Like phosphorylation of glycerol promotes non-host growth.
Yes,
and that
was one of the first PNAS paper on non-host resistance. That the glycerol one, what you are talking.
That was probably working as a signal rather than a metabolic source of a metabolic energy. So could this be also linked to some metabolic, signaling molecule rather than sucrose like metabolic energy source?
It could be. So the larger comments on the sucrose signaling was the main reason why my paper is still being dragged. So we are trying to address one by one. to systematically show it is the key role of sucrose depletion that controls the bacterial process. Everybody agrees that the bacterial population is less, more, and your concept is fine. But exactly the reason what you are asking is the one that they want more proof. So we are adding more proof in one direction, but the other direction is completely open for exploration. Like there are other substrates possible.
Great. Very good work, Sandeep.
Dr. Gopal, then Prabhu will ask. Gopal, you are asking?
Maybe actually what I was thinking that the cytokinin job Q, because it is regulating exo physiology. So maybe it is regulating how type 3 secretion system is induced during plant interaction also. So it may have role in upregulation of T2SS and T3SS, because we feel that maybe quorum sensing is also involved there in their upregulation. So maybe we'll discuss with sir later on.
Sure.
Prabhu,
can you hear me?
Yeah, we are able to hear you.
Like you are said this behavior we can expect even in soil bacteria like Mycobacterium or all these firmicutes. So can it have a role in behavior of controlling the behavior of predators, like protection from predators in soil?
No, I didn't understand. Predator means amoeba and things like that. Or what are you? What predators are you thinking about? Nematodes.
C. elegans, like there was an experiment.
Nematodes.
It ended up meeting biofilm in its front portion, like mouth portion. So something it may be doing like this soil bacteria.
So you're saying Mycobacterium may be having something like this to actually, basically in terms of the ecology with respect to the nematodes that may prey on the Mycobacteria. That's possible, Prabhu. It's a good point, actually.
So, Prabhu, actually, this point is very interesting, which you have raised. Now, people are thinking about interspecies communication also. See, Mycobacterium, when it is colonizing, so there will be many other endophytic bacteria which is there already in human, like the Bacterial or other species also. So people are talking about mixed infection and mixed biofilm formation. Is possible that Mycobacterium cytokine which is produced, cytokine like compounds which are produced by Mycobacterium may be sensed by other community bacteria which is there in the depending upon which Mycobacterium tuberculosis, like lungs and other parts they infect, and maybe some, so they might be getting the signal or something.
Yeah, actually, Subhadeep, there are also some papers which I've looked at. I've not gone through in depth. Cytokinin also modulates mammalian mammalian cells.
Yeah.
So it might actually be trying to alter its host also.
Host also, yeah.
That is one possibility. Possibility Prabhu was saying about how it may affect other predators of Mycobacterium.
That also is possible. Yeah, quite
possible. No, it's a lot of possibilities. Many things we don't know actually.
Prabhu, in your Prabhu, I wanted to. So you have done a lot of this genome analysis. So is it possible to look at those kind of predatory kind of genomes in which if there is some components which can sense those kind of cytokine-like molecule, which is
two component regulator. That
I think Subhadeep, if I understand Prabhu's question, what he is actually saying is that Mycobacterium may actually form biofilms in the mouthparts of the predator and somehow affect the predator. you know, like, and that maybe it can be cytokinin, it can be some other molecule that controls
That's quite possibly.
So that's what I think, if I understood what Prabhu was saying. Why Mycobacterium may need cytokinin, but definitely it makes cytokinin. We don't know why it does, whether it affects the host, that means humans, or it helps as some other ecological value for Mycobacterium.
Essentially, looking like not from plant side, but when there is no plant, like in soil, you don't expect any.
Sure.
It really like higher like forms of predators. And in Yersinia, the experiment was like in C. elegans, the experiment was very fantastic. Like normally it can feed on E. coli, but when they first fed this C. elegans with Yersinia, it just immediately formed in just, I think, within minutes, I think, so it made the biofilm, and then it was not able to feed on E. coli.
No, very interesting, Prabhu. Definitely. You know, I have one quick question. I have one question for Divya.
Yes.
Divya is still there. Divya?
Yeah, I'm there. Can you see me?
Yeah, it was very interesting that you've always heard about medicarpine or other phytoalexins as actually inhibiting growth of the pathogen, but you are actually saying that it has a signaling role in the infection immunity process. Has that sort of a role shown for any phytoalexin before in any plant microbe interaction? That there is a signaling role for the phytoalexin as opposed to a direct growth inhibitory role.
So yeah, people have mainly focused on the direct growth inhibitory role, but they've also shown that some of these phytoalexins, they localize to the host cell nucleus. So giving an indirect indication that they may be involved in signaling. So in our study, we were able to show that possibly it is involved in signaling. And also, if you think about plant rhizobial interactions, you have these isoflavonoids playing a very important role in this interaction. So, and a previous study had shown that, you know, when they nodulate the plants, they find that the SA levels were higher in the plant, and it was priming the plant for resistance. So that's sort of like a corroboration to our study where we found that isoflavonoids are increasing levels of SA. So it's possible that nodulation does a similar thing.
No, but bacteria, the isoflavonoid is actually serving as a regulator for the bacterium. You're saying that it's a regulator of the plant gene expression.
Yes, that is true. But we are seeing that, I mean, the other study showed that with nodulation, there was an increase in SA. They are not studying from the rhizobial point of view, but they were looking at it from the plant side of things. So maybe that interaction also results in an increase in SA. And we think somewhere that is supporting what we are seeing, where there is an induction of the SA pathway by these isoflavonoids.
The other query I had was, does medicarpine have a direct growth inhibitory role? Maybe not on your fungus, but other fungus, because I think your fungus, you can't culture it outside the...
Yes, this was actually one of the reviewer comments. They were saying, can you show it on other fungi? But there are studies which have been done where they have shown that if you treat these spores, you know, fungal spore, different fungus with medicarpine and then infect those spores on plants, they're not able to grow that well. So there seems to be a direct also impact on fungal growth.
It does both then. It has a direct...
I think it, yes, I think it does.
If I remember correctly, I think one of the oomycete, Phytophthora, is known to be inhibited by this. I hope I'm remembering correctly, but if I'm right.
Yes, you're correct. In soybean, they have shown against Phytophthora. I mean, there are many studies that have been done against different fungal pathogens and not only restricted to medicarpine, some of the intermediate compounds also, or other phytoalexins. In medicago, it's medicarpine, but in soybean, it is glycolin. It's a different phytoalexin. So they do have antibacterial as well as antifungal. And there was also a paper where a bacterial effector was found to target this pathway. So it'll be interesting to see if we can find a similar effector from powdery mildew that can actually target this particular biosynthetic pathway.
Yeah, the other point was whether you've actually looked at gene expression changes in Medicago after treatment with just Medicarpine alone.
Yes, so we did, you mean overall like genome-wide transcriptome changes? We have not done that yet. Yeah, we focus specifically on these. We did a more targeted approach where we did only with Medicarpine or after infection. But it would be interesting to see just treating with Medicarpine and seeing what happens. So can I take this opportunity to answer Dr. Roy's question? He has typed it in the chat box.
Yeah, go ahead.
Yeah, so his question is, when secondary metabolite is responsible for resistance, will it be dependent on the pathogen load? So that's a very interesting question. So what we see is that two things are important. One is the timing of the response, as well as the possibly the localization, that a local and early upregulation of this pathway seems to be important for resistance. But in the susceptible genotype, at later time points when there's a lot of fungal load, we do see induction of this pathway, but it's not able to provide that resistance. So we think that it's the timing and the localized accumulation of these compounds that would impact the growth of the pathogen. And the second part was, I think I've already answered, the resistance is to multiple pathogens. Yes, I think it can provide resistance to multiple pathogens, and other studies have shown that Medicarpine or other similar isoflavonoids can lead to resistance.
So Divya, both R108 and A17, do you see the difference, like of all the observations what you have, because the mutants are all in one? And how about the other one? Like normally these two are highly different.
They are different. Yes, it's not, they're not that similar. So that was another point, which is important to consider. But R1, the mutant collection is in R108. There's also the FNB, I think, is in
FNB is the other one,
yeah. So both can be used further to explore the role of this pathway. It's just not as easy as in Arabidopsis because we have to go through tissue culture if we are transforming or getting the mutant lines also.
Yeah.
So I have one question regarding the MIP transcription factor. Probably you overexpress one MIP gene, right?
Yes.
So there are multiple MIP transcription factors in plants, and there are R2, R3 types and different other. So how did you select this particular MIP transcription factor that are showing this biotic and working against biotic stress?
So yes, as you know, MIP transcription factor family is huge. So we narrowed down on this MIP based on our RNA-seq study, where we found that this MIP was highly upregulated in the resistant genotype, but not in the susceptible one. And it showed sequence similarity to soybean MIP. It was like a homologue of a soybean MIP, which had been previously characterized to play a role in as a positive regulator of isoflavonoid biosynthesis. So we decided to take this one. It doesn't mean that this is the only one, but to start with, we decided to pick this particular MIP transcription factor. Does that answer your question?
Yes, but is there any specialty that is R2, R3 type, R2, R3 type, or any other kind?
It's an R2, R3 type.
Okay.
Yeah.
There is one more question for Dr. Divya.
Mm-hmm.
If salicylic acid is also induced during nodulation by rhizobia, then how rhizobia able to multiply inside plant during defense? Asked by Shraddha Dahel.
A very good question. So in that study, they found that SA levels increased in the leaves, not in the roots, but the SA levels were increased in the leaves, and then they infected with powdery mildew, and they found that it was able to enhance resistance. Now, whether it also increases, I can't recall right now whether they looked at the root, but I remember that they definitely looked at the leaf tissue. Not sure about the root, but a good question.
Question for Gopal ji.
Yeah.
Yes, Prabhu.
Is Dr. Gopal?
Yeah, please. Please go ahead, Prabhu.
Gladiola.
Prabhu, go ahead.
You are using Burkholderia gladiola as model system, na? Like model bacteria.
Yes.
Your hypothesis that this defensive toxin antitoxin system is converting into offensive T6SS effector.
Yes.
Or do you think it is happening at the genus level, or you have population level studies in gladiola itself and make it like see if it is happening only in this species because
Actually,
of its
yeah, shall I add on actually? So what we observed is, as I've showed you the global phylogeny, so in bacteria, the diverse bacteria, which encode either them as TA or T6SS. So it's not just specific to Burkholderia, even in actinomycetes bacteria and things like that. Most of the actinobacteria, it was encoded as TA. But in gamma proteobacteria, it was encoded both as TA and T6SS. So mostly we figured out this presence of IS elements in Burkholderia species. So in that sense, you can say it is specific to Burkholderia, this evolution. But then there is Pseudomonas actually, Pseudomonas aeruginosa. So there is Pseudomonas aeruginosa POA strain. There they have characterized the same antitoxin system and shown it as a T6SS effector. There also we figured out in some Pseudomonas strain, they are only encoded as TA, and while others, they are encoded as both TA and T6SS. So I think we have evidence for both Burkholderia and Pseudomonas, but the rest of them, we don't know whether conversion is happened or they have selected it and keeping it.
Okay.
So Gopal, is that RNAs, RNAs, there are different types of RNases, right? So which one is it most similar to, like the T family, F family, A family?
H family.
H family.
I think we can encourage the students also to ask questions if they want.
Yeah. If any question is there, sir.
So Indra, I think there is one question in the chat box. Student, I think.
So I think Subhendra was interested to know RNA is any sequence specific.
I think yeah, that is Divya madam asked that question about RNase H.
Yeah, so it's right now we don't know it's any specificity because it is killing both, degrading both pre-bacterial RNA as well as the host own RNA. So we have not seen any specificity.
So Dr. Amit has asked to all, can we comment on field application of secretory compounds for bacterial and fungal disease control?
Okay, maybe I'll try and take this, Subhendra.
Yes, sir. Yes, sir.
Amit, there are experiments where people have done, they've used something called harpin. Harpin is a type 3 secretion system secreted protein that induces hypersensitive response. So there are, I think it was pioneered by scientists at Cornell University, and they showed that application of harpin can induce field level resistance. I'm not sure that will work with ZOPQ, it's not going to work. But proteins that can induce a hypersensitive response, and harpin I think was a small protein. So they had been using it, but I've not really heard much about it in terms of its application in a wide scale or widespread use of this. So there may be limitations in terms of taking the secreted protein like a harpin. But maybe Divya can talk about the medicarpin or some others as antifungal agents.
Yeah, I mean, that was the goal of doing this study, to see if we can use any of these, compounds for fungal disease control and maybe even bacterial disease control. But I think right now there's also parallel research going on as to how to increase the production of these compounds, because to synthesize these it's quite expensive. So maybe to increase the production or change the metabolic flux of some of these pathways, or the plant itself can produce more of these compounds, and then we can grow those plants in the field. So that is my take on this particular aspect.
And Gopal can maybe talk about his protein.
Yes. So actually, as I said, we have identified a profile tail-like protein, which is antifungal. So we try to spray this protein on rice, and it is protecting against disease, particularly seed fly disease. And our unpublished work is, though it's very exciting, it's very preliminary right now. We are coming up that this protein may also be inducing the defense response. And we have come up a smaller peptide now. So that peptide, we are in the process of receiving it. If it starts inducing defense response, maybe that peptide may be more suitable for field application. That's what we are anticipating. And problem is actually the protein-based spray in rice is little difficult, what we have realized it, because rice is grown under the watery condition. And once we spray and rain comes, there is no fun. So somehow these things has to go inside, and then it may be more durable. So maybe with peptide, I think we can evolve some things so that it can be systemic, go inside and induce the defense response. So it's a great open field, exciting field. Little bit we are working, but with protein we are facing limitation.
So Gopal, can we use nanoparticles maybe to assist?
That's what. So those things we are now developing.
Also, his protein we are expressing for the dry root rot. Initial, I think you can explain that.
Yeah. So maybe one of our students will talk to next day. So maybe we can hear to him. Yeah. So it is even protecting. Actually, thanks to Ramesh sir, he encouraged both of us to collaborate and see whether it is controlling dry root rot, and indeed we are coming up that it is maybe a good solution for dry root rot. So one of the students will make a detailed presentation later.
I have to log off now.
Sir, there is no more question.
Yeah.
So at 4 o'clock again, sir.
Sure. We'll meet at 4.
Sir, we will conclude now.
Yeah.
This session.
Sure.
So I think, sir, we are.
Thanks, Sovendra. It's a great job. And actually, congratulations and well appreciation to all the speakers.
It's wonderful.
Yeah, wonderful session. And thanks to all for.
So we are joining at 4 o'clock.
At 4 p.m. Yeah.
4 o'clock. Okay.
Thank you. Namaskar.
Bye-bye.
Bye.
हेलो सुवेंद्र चले गए?